right. Last lecture. I'm sure we're all just jumping out of our seats right now. So, we're going to talk about reproduction and development. All right. So, now when you're looking at human reproduction, okay, we produce offspring via sexual reproduction. Okay. And that's where we have a haploid sperm that's produced by the testes of the males, and it goes and it fertilizes the haploid secondary oocytes that are produced by the ovaries of the females. So, as a result of fertilization, the two haploid cells come together, they form a diploid cell. That's called a zygote. It contains one set of chromosomes from each parent. So I get your mom and your dad's characteristics. Okay. Now, males and females have anatomically distinct genitalia regions. I'm sure we're all aware of that, but they're all there to produce, nourish, and transport haploid cells to basically get to these points that we need to be in, where we're having fertilization, we're growing the embryo and the fetus. Okay, and all genitalia or organs, the male and a female, they can be grouped by function. Right, the gonads, testes in males and ovaries in females all produce gametes. They secrete sexual hormones. Various ducts that store and transport the gametes and whatever accessory male genitalia glands, which produce substances that protect the gametes until they have to do their movement, and supporting structures like the penis, the uterus in the females. Okay, all that are there to assist delivery of the gametes. And the uterus is also the site for the growth of the embryo fetus during pregnancy. So, as expected, quite important. Now, the male reproductive system. Okay. Now, starting off, you know, it obviously has some key functions, right? Producing sperm, right? Spermatogenesis, transporting and delivering that sperm, producing whatever seminal fluid is needed to produce and carry such sperm. But it's also there to help with secreting testosterone. Okay, now the testes are what produce the sperm, okay, and testosterone. Now, the ducts that are associated with all this, they help to transport, store, and assist the maturing of the sperm. Accessory male genitalia glands, they are the guys that secrete most of the liquid portion of semen that it's commonly associated with. And of course, the penis contains urethra. That's passageway for ejaculation of semen and excretion of urine. Okay, now the organs include all of that, right? Testes, systems of ducts, which we'll get through, such as urethra, ejactorial ducts, epididymis, accessory male genitalia glands like seminal glands, prostate glands, as well as a couple other supporting functions like the scrotum and the penis. That's the entire male genitalia system, right? Like we just said, testes are the male gonads. They're the ones that produce the sperm and secrete the hormones. Duct systems is what gets that all there and also helps to store the sperm until it's matured and ready to go. All right. Now, the scrotum, okay, it's a supporting structure specifically for the testes. And it consists of loose skin, okay, and underlying subcutaneous tissue that hangs from the root, okay, specifically the root of the penis. Externally, the scrotum looks like a single pouch of skin separated into lateral portions by a median ridge that's called the raphe. Internally, the scrotal septum divides the scrotum into two compartments. Each one of those contain a single testicle. The septum is made up of subcutaneous tissue and muscle tissue, which is actually called the dortus muscle. And it's composed of smooth muscle fibers. Now, this muscle is also found in subcutaneous tissue of the scrotum. And associated with each one of these testes in the scrotum is known as the cremaster muscle, which is just basically a bunch of skeletal muscles that form in small bands that descend as an extension of the interabdominal muscles through the spermatic cord to surround the testes. Okay. The location of the scrotum, okay, and the contraction of the muscle fibers regulate the temperature of the testes. So For instance, normal sperm production requires temperature of about 2 to 3 Celsius, okay, below our core body temperature. Lower temperature is maintained within the scrotum because it's outside the pelvic cavity. So in response to cold temperatures, the muscles contract. Okay, and contractions of these cremaster muscles that moves the testes closer to the body, and that's because they can then absorb the body heat that's being emitted. Now, contraction of the other muscles causes scrotum to become tight, wrinkled in appearance. And this actually goes and reduces heat loss. All right, so, and obviously, if you'd expose it to warmth, it reverses all these actions in the opposite direction. Okay, but all done in a way to optimize spermatogenesis by controlling body heat, absorbance, and secretion. Now, the testes are paired oval glands in the scrotum. Now, when I'm saying testes, that's singular, testicles are plural, obviously. They're quite small, okay, like 15-ish grams on average, maybe a little less. But they develop near the kidneys, a posterior portion of the abdomen, and they will eventually begin to descend into the scrotum through what are known as the inguinal canals, which are just passageways in the lower anterior abdominal wall. And this occurs during the later half of the 7th month of male fetal development. Histologically, these contain a serous membrane called the tunicle vaginalis, which is derived from peritoneum, and it forms during the descent of the testes, actually, and it partially covers the testes. Now, internally to the tunica, the testes is surrounded by a white fibrous capsule that's composed of dense, irregular connective tissue, which is known as the tunicula appendae. And it extends inward, and forming the septa that divides the testes from a series of internal compartments that are called lobules. Each of the around 300-ish lobules contain 1, 2, 3 tightly coiled tubules, the seminiferous tubules. This is where sperm is produced. Okay, and the process that follows, that occurs at this point to make sperm, is known as spermatogenesis. Looking a little closer into those tubules, the seminiferous tubules, they contain two types of cells, spermogenia and sperm-forming cells in nursing cells. They have several functions which actually help to go and support spermatogenesis. Now, spermatogonia, these are stem cells, okay, specifically stem cells that have developed from primordial germ cells. And they arise from the umbilical vesicle that then enters the testes. And that's around the 5th week of development, male fetal development. In the embryonic testes, the primordial germ cells differentiate into spermatogones, which remain dormant during childhood and actively begin producing sperm at puberty. Towards the lumen of these tubules, there are layers of progressively more mature cells. All right, so we're going more and more mature. In order of advanced maturity, there are primary spermatocytes, secondary spermatids, and sperm. So after a sperm or a spermatozoon is formed, it's released into the lumen of the seminiferous tubes and sperm's made. Now, embedding among the spermagena cells in the seminiferous tubes are large nerve cells. Okay, they're also known as subsyntecular cells or cerebrate cells. I'm going to call them nerve cells. These guys extend from basement membrane to the limit of the tubule and internal to the basement membrane and the spermagena. Tight junctions join these neighboring nerve cells to one another. These junctions form an obstruction that's known as the blood-testes barrier. And it's because of substances that must first pass through the nerve cells before they can reach the developing sperm. Okay, so it's almost like a little barrier, protective security guard. So by isolating the developing gametes from the blood-testes barrier prevents an immune response against the surface antigens of spermatogenes, which those guys are actually recognized as foreign by the immune system. Okay, now nurse cells support and protect developing spermagena cells in several ways. Right, they either nourish, all right, so the spermicide, spermatid, sperm, They phagocytize excess spermatid cytoplasms as development will proceed. They control the release of sperm into the lumen of the tubules, and they also produce fluid for sperm transport, so like hormone inhibin, okay, regulate the effects of testosterone, FSH or follicle stimulating hormone. In the spaces between adjacent and seminiferous tubules and clusters of cells called interstitial endocrine cells or Lidwick cells, these areas where the hormone testosterone begins to be secreted, most prevalently as an androgen. Now, the androgen is a hormone that promotes the development of masculine characteristics. Okay, so this is where you get the deeper voice, testosterone promoting the man's libido. These are the key players of when puberty hits, when you start to see the changes. So in humans, when we go through spermogenesis, it only takes around 60 days. Okay, and it begins with the spermatogen, and that contains two N chromosomes, so diploid. Okay, now these guys are stem cells, and when they undergo mitosis, some of them remain near the basement membrane of the seminiferous tubes and in an undifferentiated state. And that's because they want to serve as a reservoir of cells for future cell division, subsequent sperm production. Now, the rest of them, they lose contact with the basement membrane. They squeeze through the tight junctions of the blood testes barrier, and those then undergo developmental changes and differentiate into primary spermatocytes. Now, those guys, like the stem cells, spermatogenes, are diploids. Okay, so they have 46 chromosomes. Now, shortly after it forms, each primary spermatocyte is going to replicate its DNA, and then meiosis begins. Now, in meiosis 1, homologous pairs of chromosomes line up at the metaphase plate and crossing over occurs. That's when the meiotic spindle pulls one duplicated chromosome of each pair to an opposite end of the dividing cell. The two cells formed by meiosis 1 are called our secondary spermatocytes. And now each secondary spermatocyte has 23 chromosomes, right? Each chromosome within a secondary spermatocyte, however, it's made up of two chromatids and still attached by their centromere. No replication of DNA occurs in these secondary spermatocytes. Now in meiosis 2, the chromosomes line up in single file along the metaphase plate, and the two chromatids of each chromosome separate. The four haploid cells result from meiosis 2, and they become known as spermatids. A single primary spermatocyte can there go forth and produce four spermatids with the haploid number. Okay, and that's all by 2 rounds of cell division, meiosis 1, meiosis 2. The unique process occurs during spermogenesis, though. So as sperm proliferate, they fail to complete cytoplasmic separation, cytokinesis. So the cells remain in contact via cytoplasmic bridges through the entire development. Now, this pattern of development, it most likely accounts for synchronized production of sperm in a given area of the seminiferous tubes. It may also have survival value to the half-life of the sperm contained an X chromosome and half contain a Y chromosome. The larger X chromosome may carry genes needed for spermatogenesis that are lacking on the smaller Y chromosome. Now, as we go through spermatogenesis, the final stage, or spermatogenesis, is the maturation of the haploid spermatids into sperm. Now, no cell division occurs in spermatogenesis, and each spermatid becomes a single sperm. During this process, spherical spermatids transform into elongated, slender sperm, right? An acrosome forms atop the nucleus, which condenses and elongates a flagellum that develops, and a mitochondria then multiply. Nurse cells will then go through and they'll dispose of any excess cytoplasm that kind of sloughs off, falls off as it's going through the process. And then lastly, sperm are released from their connections to the nurse cells, and that is an event known as spermination. And sperm then goes and enters the lumen of the seminiferous tubules, and fluid secreted by nurse cells pushes sperm along the way for mobility the towards of the ducts of the testes. Okay. At this point, though, sperm is not yet able to swim. Now, this is just a kind of close-up picture of exactly what I'm talking about when I'm talking about the structure of the sperm. It's associated with the head and the tail. You kind of commonly get that little S-shaped body. And so each day, we talk about spermogenesis. It's like 300, 200 million sperm a day can be made just by doing spermatogenesis. And they're really small, 60 micrometers long. Right? They got several structures, and it's all highly adapted and all about having its capability to reach and penetrate an egg for fertilization to occur. Okay? And the major parts of the sperm are its head and tail, the flattened, pointed head of the sperm, and that's what's going to go, and it's going to have the nucleus, okay, with those 23 highly condensed chromosomes, and it's going to be covered by the anterior two-thirds of the nucleus, that is the acrosome. And that's just a cap-like vesicle that's filled with enzymes that actually help the sperm penetrate into the egg, right, to bring about the fertilization. Among the enzymes, those guys are hyaluronidase and proteases. That's the two big guys that are involved. Okay. The tail of the sperm, so the other half, okay, is subdivided into four parts. We got the neck, we got the middle piece, principal piece, and the end piece. The centrioles from the microtubules that comprise the remainder of the tail. The neck is the constricted region that behind the head contains the centrioles. And then the middle piece contains the mitochondria arranged in a spiral. And that's what is actually giving the ATP energy needed for its locomotion, or the wiggling of the tail you've probably associated it with. And so this locomotion is what helps them get to the site of fertilization. And it actually helps them with their own metabolism, which is called sperm metabolism. Now, the principal piece of this all is the longest portion. That's the tail, longest portion of the tail. And the end of this is the terminal, and the tapering portion of the tail, which kind of is quite important for mobility, getting to where it needs to be. Now, all no initiating factors are unknown. At puberty, we get a hypothalamic neurocirculatory cell increase, specifically in their secretion of gonadotropin releasing hormone, or GnRH. This hormone stimulates gonadotrophic cells in the anterior pituitary to increase the secretion of two gonadotropins: luteinizing hormone, or LH, follicle stimulating hormone, FSH. Now, LH stimulates interstitial endocrine cells, which are located between the seminiferous tubes. This then allows them to secrete the hormone testosterone. This steroid hormone is synthesized from cholesterol in the testes and is the principal androgen that is used. Okay. It is lipid soluble and readily diffuses out of our interstitial cells and into the interstitial fluid and then into the blood. Okay. Now, via negative feedback, testosterone can suppress secretion of the LH by anterior pituitary gonadotrophs and suppresses secretion of that GnRH by the hypothalamic neurocirculatory cells. In some target cells, such as external genitalia, the prostate, a specific enzyme can convert testosterone into another androgen. That's called dihydrotestosterone, or DHT. Now, FSH acts indirectly to stimulate spermatogenesis. FSH and testosterone act synergistically, actually, on nerve cells, and it stimulates secretion of androgen binding protein, or ABP, specifically into the lumen of the seminiferous tubules, and then into the interstitial fluid around the spermatogon. Now, ABP goes and it binds to testosterone, and it does it to keep its concentration high, and then testosterone goes forth and stimulates the final steps of spermatogenesis in the seminiferous tubes. Once the degree of spermatogenesis required for male reproductive functions is achieved, nerve cells can release inhibin, and that's a protein hormone named for its role. It inhibits FSH secretion, specifically by the anterior pituitary. Now, if it's going too slow, if spermatogenesis is just proceeding too slowly, less inhibin is then released, and that promotes more FSH secretion and an increased rate of spermatogenesis. So again, it's about balancing these two. Looking at testosterone and that extra form of testosterone, dihydrotestosterone, both of those can bind to androgen receptors. Specifically, they can bind to the same androgen receptor. They're all found within the nuclei of target cells. And this hormone receptor complex is what helps regulate gene expression. Okay, so it helps us to turn some genes on, turn some genes off. And because of this, androgens can have some significant effects. Now, for instance, prenatal development. Before birth, testosterone just stimulates the male pattern of development of genitalia and the system ducts associated with it, as well as when the descent of testes is going to occur. Whereas the dihydrotestosterone, that stimulates the development of the external genitalia. Testosterone also is converting in the brain and estrogens, which basically plays a role in development of certain regions of the brains in males. Now, obviously, these can come into play with physical characteristics of developing male sexual characteristics as well. Um, increase of libido, having secondary sex characteristic traits. This change of the muscular skeletal growth, widening shoulders, narrowing hips, facial and chest hair. Again, all led into effect with these two main forms of testosterone playing a role. Testosterone is obviously extremely important. It controls a whole lot of stuff. So it has to be controlled, right? And one of the main ways it's controlled is a form of negative feedback. Okay, and so when testosterone concentration in the blood is increased to a certain level, it actually goes and inhibits the release of that GnRH by cells in the hypothalamus. So because of that, there's less of that GnRH in the portal blood that flows from the hypothalamus to the anterior pituitary. Now, gonadotrophic cells in the anterior pituitary, they release less LH. So the concentration of LH in the systemic blood falls. And with less stimulation by that, the interstitial endocrine cells in the testes secrete less testosterone, and then it becomes a return to the homeostasis. Okay, and if the testosterone concentration of the blood falls too low, however, GnRH is again released by the hypothalamus and stimulates secretion of LH by the anterior pituitary. And then LH in turn stimulates testosterone production by the testes further to promote homeostasis going in the opposite direction. Now, there's a lot going on in this entire process. Okay, and all the cells coming together and communicating with one another. And a lot of it is led by one cell, and it's the nerve cells. So the nerve cells are secreting fluid. Okay, and that's all because they're trying to push sperm fluid along the way. But in the process, they're generating pressure. And this combined with one another pushes those fluids along the lumen of the seminiferous tubules and then into short ducts called straight tubules, and then even further into a system of ducts that are known as the red testes, and then lastly all the way into these coiled afferent ductules. Now, as it goes through this pathway, it gets to the epididymis. This guy is decently small organ. It's pretty long, curves kind of similar to a comma shape, and it's on the border of each testis. Now, this guy connects with what is known as the duct of the epididymis. Okay, and it's basically a superior portion of the epididymis, which is known as the head, and the body of the epididymis as a narrow portion into the tail. And the tail is then where it connects into the duct. Okay. Now the duct, if it was uncoiled, decently long, like 10 feet long. It's pretty crazy. Now, however, it is coiled, and it's actually lined with pseudostratified columnar epithelium, and it's encircled by a layer of smooth muscles. Now that pseudostratified columnar epithelium, we've kind of seen some of this before in a couple other chapters, but the free cells are containing stereocilia. Now it's not cilia, it's actually microvilli, right? But they're there to do the same exact thing, and that is to increase the surface area of these cells to help with reabsorption. Now, besides that, it's got a lot of connective tissue around it, and it helps to connect it to a muscular layer, as well as keep everything in place that it needs, like blood vessels, nerves that are needed, things like that. And functionally, this guy, the epidermis, is pretty important. Okay, so you got all the spermogenesis going on before this. However, just because we got sperm made doesn't mean it's mobile, doesn't make it fertilize. When it gets to the epidermis, it can. Okay, it acquires its mobility, and as such, its ability to fertilize. Okay, and this occurs like every 14 days. Now, it's got some other things that's important with it. It helps with propelling the sperm during sexual arousal. That's because of contraction of the smooth muscles surrounding it. It also can act as a storage center. Okay, and you can even keep sperm there for months, still be viable. So at the end of this system, right at the tail of the epidermis, it starts to change its shape. Its diameter begins to increase. It looks a little less convoluted, and that's at this point it's known as a ductus deferens. Okay, now this guy is long, and it ascends along the posterior border of the epidermis through the spermatic cord and enters the pelvic cavity. Once it's in their pelvic cavity, loops over the udder and passes over the side and down the posterior surface of the urinary bladder. Okay. The mucosa of the ductus deferens consists again of that pseudostratified columnar epithelium as well as that connective tissue. Okay, now difference here is the muscular layer is a little more grown. It's got 3 layers this time, with the inner and outer layer being longitudinal smooth muscle and the middle layer being circular smooth muscle. Now, very similar function to the prior. It's mostly there to help with keeping the flow of the sperm, especially during sexual arousal, but also can act as a storage hall for sperm if needed as well. Now, the spermatic cord, okay, that's a supported structure of the male genitalia system. So that ascends out of the scrotum. And each one of those cords consists of one of those ductus deferens. And that's as it ascends through the scrotum, the testicular artery, the veins that drain the testes and carry testosterone in circulation, our autonomic nerves, lymphatic vessels, and muscles. Okay, a whole lot going on. The spermatic cord and ileoinguinal nerve passes through our inguinal canal. Okay, that's just an oblique passageway that's in the anterior abdominal wall that's just superior and parallel to the medial half of our inguinal ligament. All right, that's pretty small canal. But it's pretty important when it comes to assisting with the flow of the pressure. And there are basically comparisons of this when we get to the female anatomy that function as the same way. So, one of the main kind of things with the male genitalia system is ejaculation, right? How else are we going to get to fertilization if we don't have these haploid cells coming together? And this is when ejaculatory ducts comes into play. Okay, so ejaculatory ducts are long. They're about long in relative terms. They're about an inch. Okay, and it's formed by the union of a duct from our seminal glands, which we'll talk about the glands in a bit, and our ductus deferens coming together. Okay, and these ejaculatory ducts, they form just superior to the base or superior portion of the prostate, and they pass inferiorly and anteriorly through the prostate. And they terminate in the prostatic urethra. And this is where they eject the sperm and seminal gland secretions just before the release of semen from the urethra to the exterior, outside. And we'll get to more specifics on that in a bit. But as I just said, seminal glands, we have accessory genital gland, and they each have their own kind of secretion that plays its role. Okay. Now, the seminal glands, they secrete seminal fluid. That's an alkaline, viscous fluid that helps neutralize acid in the female genitalia tract. It also provides fructose for ATP production by the sperm and contributes to sperm motility and viability, as well as it helps semen coagulate after ejaculation. The prostate, it secretes prostatic fluid, which is like a milky, slightly acidic fluid, and has enzymes that commonly will break down clotting proteins from seminal glands. The bulbourethral glands secrete alkaline fluid that neutralizes the acidic environment of the urethra and mucus that lubricates the lining of the urethra and the tip of the penis during sexual intercourse. Okay. Now, the urethra. Okay, so that's kind of like the final tunnel, right? And in males, it's just the shared terminal duct of both our genitalia and our urinary systems. It's just that finishing pathway for semen and urine. Now, this prostatic urethra is 2 to 3 centimeters long, right, and it passes through our prostate. And as this duct continues inferiorly, it passes through the deep muscle of the perineum. This is where it's known as a membranous urethra. The membranous urethra is pretty small. It's about like half an inch. But as this passes through the corpus sphincter of the penis, it's known as the spongy urethra. And that is much larger, 6 to 8 inches. The spongy urethra ends at the external urethral orifice. Okay, so the urethra changes by its location. Okay, and the tissue as such that is surrounding. All right, such as the three parts of prostatic urethra, membranous urethra, and our exterior. Now, looking a little deeper into our genitalia glands, okay? Now, the main accessory male genitalia glands are seminal, prostate, and the bulbourethral glands. So we talked a little bit about them in the previous slide, but the seminal glands, they're paired. Okay, they're paired glands, okay, and they're convoluted pouch-like structures. Now, through the seminal gland ducts, the seminal glands secrete seminal fluid. Now, if you remember, that's that viscous alkaline solution. Sometimes it's got fructose, prostaglandins, even has some clotting proteins, however, proteins that are different from those in blood. And that whole solution is beneficial. Okay, alkaline nature of the seminal fluid actually neutralizes the acidic environment of the male urethra and female genitalia tract. Okay, because that would either kill or inactivate your sperm if it came in contact. The fructose is used for ATP production by sperm. Prostaglandins contribute to sperm motility and viability. They can even stimulate smooth muscle contractions within the female genitalia tract. Clotting proteins help semen coagulate after ejaculation. Okay? Now, the prostate. The prostate is probably one of the ones I'm most focused on because of its role in cancer. But the prostate is just a single donut-shaped gland, and that's about the size of like a golf ball. Right? So it's actually not that big. Okay, it is inferior to the urinary bladder and surrounds the prostatic urethra. The prostate slowly increases in size from birth to puberty. So as such, you would expect it's got some type of role, some hormone interaction, and it secretes with prostatic fluid. Okay, and that's a milky, kind of slightly acidic solution that has several substances. Okay, it has citric acid, right? That's for ATP production. It has proteolytic enzymes. Okay, that's to help break down clotting proteins that are inside the seminal glands. It has some other things that aren't even known, or what their function is known. So, like acid phosphatase, it's secreted. They don't know what that's there, but it's there. Seminal plasmin, okay, is an antibiotic that destroys bacteria, right? And it can actually help decrease the number of natural occurring bacteria in semen and lower it in the female genitalia tract. Okay, so we know a lot about what secretions do what, but we also don't know too much about them at the same time. Now, the last one, bulbourethral glands. Okay, these guys are about the size of peas, and they're paired. There's two of them. Okay, and they're located inferior to the prostate on either side of the membranous urethra within the deep muscles of the perineum. During sexual arousal, these glands secrete an alkaline fluid into the urethra that protects the passing sperm by neutralizing acids from urine and urethra. They also secrete mucus that lubricates the end of the penis and the lining of the urethra and decreasing the number of sperm damaged during ejaculation. So this whole thing that I've got to so far, the main player seems to be semen. All right. So looking at semen, it's different than sperm. Okay. Sometimes common misconception is they're the same thing. It's not. So semen is actually a mixture of sperm with seminal fluid. Okay, a liquid that consists of secretions of seminiferous tubules, seminal glands, prostates, other glands, is all those mixtures together. That is semen. Okay, it's not one or the other. Now, despite slight acidity of prostatic fluid, semen still has a slightly alkaline pH. Okay, that's due to the higher pH and larger volume of liquid from the seminal glands. The prostatic fluid gives semen its milky appearance, and fluids from the seminal glands and the bulboreal glands give it its more sticky consistency. And okay, so what the hell's the point of seminal fluid, right? Well, it's because it's there to support it. It's there to help it move. It's like a transportation medium. It's there to give it the nutrients like the sugars for the ATP to help them move, as well as there for protection against any acidic environment, whether it be inside the male's urethra or the female's vagina. Okay, but a lot of it changes as it goes through and as it goes out of the body. So once ejaculated, liquid semen coagulates within 5 minutes, and it's all because there's clotting proteins within this solution, okay, specifically from the seminal glands. And the functional role of semen coagulation, it's not really known. Okay, but we know there's proteins involved that are different from those that cause blood coagulation, so it's not the same exact thing, it's not the same exact process. After about 10 to 20 minutes, it reliquefies. And it's because of the prostate specific antigen, PSA, or some other proteolytic enzymes that's produced by the prostate. It starts to break down the clot. Now, abnormal or delayed liquefaction of clotted semen can cause complete or partial immobilization of sperm, so inhibiting the movement through the cervix of the uterus. Okay, so after passing through the uterus and uterine tube, the sperm are affected by secretions of the uterine tube. So that's a process known as sapatiation. So the presence of blood and semen, unfortunately, is what is known as hemospermia. And a lot of times it's when stuff like this goes wrong. All right, there's inflammation within the tracts that semen is being produced in, which is leaking into the blood vessels, causing some blood or a track of blood into the pathway that semen or sperm is following. So the penis, it's part of the spongy urethra, okay? And it brings in the urethra, which is just that single passageway for ejaculation or excretion of urine, okay? It's the cylindrical in shape, and it consists of a body, glans, penis, and a root. Okay, the body of the penis is composed of three cylindrical masses of tissue, each surrounded by dense, irregular connective tissue, which is called the tunica albuginea. And the two dorsolateral masses are called the corporal cavernosa penis. Okay. The smaller midventral mass, the corpus spongiosum penis, contains the spongy urethra and keeps it open during ejaculation. Skin and the subcutaneous tissue enclose all three masses, okay, which consist of erectile tissue. Now, erectile tissue is just composed of numerous blood sinuses or vascular spaces, and it's lined by endothelial cells and surrounded by smooth smells as well as elastic connective tissue, which makes it stretchy. The distal end of our corpus spongiosum penis is a slightly enlarged acorn-shaped region that's called the glans penis, and its margin is the corona. The distal urethra enlarges when the glans penis and forms a terminal slit-like opening, the external urethra orifice. Now, covering the glans is an uncircumcised penis is the loosely fitting prepuce or foreskin. Um, Mostly it is removed during circumcision, however, not always. Now, the root of the penis is the attached portion, or the proximal portion. It consists of the bulb of the penis, the expanded posterior continuation of the base of the corpus spongiosum penis, and the groove of the penis. The two separated and tapered portions of the corpus crepinus penis. The bulb of the penis is attached to the inferior surface of the deep muscles of the perineum and is enclosed by the bulbospongious muscle, a muscle that aids in ejaculation. Now, each of the penis bends laterally away from the bulb of the penis to attach the ischial and inferior pubic rami and is surrounded by the ischiocavernous muscle. Now, the penis is actually supported by two ligaments that are continuous with the fascia of the penis. The fundiform ligament that arises from the inferior part of the linea alba, and the suspensory ligament of the penis, which arises from the pubic symphysis. Upon sexual stimulation, parasympathetic fibers from the sacral portion of our spinal cord initiate and they maintain the erection, okay, or the enlargement and stiffening of the penis. The parasympathetic fibers produce and release nitric oxide. So that nitric oxide is what causes smooth muscle in the walls of the areolas, supplying the erectile tissue to dilate, okay, or relax. So that in turn goes and it causes large amounts of blood to enter the erectile tissue of the penis, and nitric oxide also causes the smooth muscle within the erectile tissue to relax, resulting in widening of the blood sinuses. This combination of increased blood flow and widening of the blood sinuses, that's what results in an erection. Expansion of the blood sinuses also compresses the veins and drains the penis. So the slowing of blood outflow actually helps to maintain the erection. Now, then the insertion of an erect penis into the vagina, that's called heterosexual intercourse, okay, or coitus. So a major stimulus for the erection is, in fact, this mechanical stimulation of the penis. And it coincides with mechanoreceptors. So mechanoreceptors will provide direct input to the erection, integrating center in the spinal cord. So erotic sights, sounds, smells, thoughts can stimulate this. Okay. And this involves descending inputs from the brain, so like the hypothalamus, the limbic system, to the spinal cord. And negative stimuli is also shown to have effects. So, like in a bad mood, depression, anxiety, it can inhibit these pathways and, as such, inhibit the erections. Now, there are times when this can be messed up, okay, this whole entire regulated system, and we get some unfortunate disorders such as peripasia, which is just a persistent and it's a very usually painful erection of the penis and doesn't just simply involve that similar pathway that correlates with sexual desire or whatever excitement. Okay, this can go from anywhere from hours all the way to, unfortunately, some days. Okay, and it does result in some intense abnormalities like blood vessels, nerves, things like that. So the whole point of coitus and all that is to get to a point what's known as ejaculation, okay, or the powerful release of semen from the urethra to the exterior. Okay, it's a sympathetic reflex that's coordinated by the lumbar portion of the spinal cord. So as part of the reflex, the smooth muscle sphincter at the base of the urinary bladder closes, so that prevents urine from being expelled during ejaculation and semen from entering the urinary bladder, so it kind of acts like a barricade off, blocking off the pathways. And so even before ejaculation occurs, peristaltic contractions in the epididymis, ductus deferens, seminal glands, ejaculatory ducts, and prostate is what propels the semen into the spongy urethra. So typically this leads to emission, okay, or the discharge of a small volume of semen before ejaculation. Emission may also occur during some other times, okay, like sleep, known as nocturnal emissions. Now, the musculature of the penis, the bulbus spongius, and the superficial transverse perineal muscles. This is what's just supplying the penile nerves and contacts at the ejaculation. So once sexual stimulation of the penis is ended, the areolas supplying the erect tissue of the penis constrict, smooth muscle within the erectile tissue contracts, which then makes the blood sinuses smaller. And this basically relieves the pressure on the veins, supplying the penis with the blood, and it allows it to drain through. So at this point, the penis returns to its flaccid or relaxed state. Now, moving from male to female reproductive system, the female reproductive system is responsible for a lot of stuff and a lot of important things, especially when it comes to development. Okay, such as producing oocytes, supporting fertilization, maintaining the pregnancy, enabling childbirth, providing nourishments. And this entire system includes a lot of supporting structures. Okay, the ovaries, also known as female gonads, uterine tubes, oviducts, the uterus, the vagina, all the external organs that come with it, including mammary glands, for instance. While mammary glands are integumentary systems, they are also actually considered a part of the female genitalia system. Now, ovaries, or female gonads, you may hear them called, I'll call them ovaries. These are paired glands that resemble kind of almond shape, and they're actually similar size as well. And they're pretty much homologous to the testes. Okay, so the ovaries produce a couple things. One being the gametes, or secondary oocytes that develop into mature eggs after fertilization, and hormones, so like progesterone, estrogen, inhibin, relaxin. The ovaries, one on either side of the uterus, descend to the brim of the superior portion of the pelvic cavity during our third month of development. So a series of ligaments holds them into that position. The broad ligament of the uterus, which is folded by the paraperitoneum, attaches to the ovaries by a double layered fold of the peritoneum called the mesovarium. And the ovarian ligament anchors the ovaries to the uterus, and the suspensory ligament attaches them to the pelvic wall. So each ovary contains what is known as a hilum. And this is the point of entrance and exit for all the blood vessels, all the nerves, okay, along with that mesovarium coming in. Now, looking further into the tissue and the histology of the ovary, it consists of a variety of parts. Okay, the ovarian mesothelium or surface epithelium is the simple layer of epithelium that covers the surface of the ovary. It's commonly low cuboidal or squamous. Okay, the tunica linea is kind of this whitish capsule of dense, irregular connective tissue that's immediately underneath that mesothelium, okay, the ovarian mesothelium. And then the ovarian cortex, that's a region just deep to the tunica linea, and it consists of ovarian follicles surrounded by dense, irregular connective tissue, which contains collagen and fibroblast like cells that are called stromal cells. Now, the ovarian medulla is deep to the ovarian cortex, and the border between the cortex and medulla, that's pretty much indistinct. It's not really a border, but the medulla consists of more of this loosely arranged connective tissue, which contains like our blood vessels, lymphatic vessels, nerves. Ovarian follicles, okay, they are in the cortex, and they consist of oocytes. And the oocytes consist in various stages of development, okay, plus all those cells that surround them, all right? These oocytes are immature egg cells, okay, and when the surrounding cells form a single layer, they become what are known as follicular cells. And later in development, they will eventually form several layers, and these guys are referred to granulosa cells. Okay, the surrounding cells are there to basically nourish any developing oocyte, begin to secrete hormones like estrogens as follicles grow larger in the ovary. Okay, and a tertiary ovarian follicle is also known as a vesicular or Grafton follicle. I'm going to call it an ovarian follicle. These guys are quite large. All right, and they're fluid filled. It's basically almost ready to rupture, okay, and expel its secondary oocyte. Okay, this is a process known as ovulation. Okay, and usually around this time after ovulation, corpus luteum contains the remnants of these tertiary ovarian follicles, okay, after ovulation. And the corpus luteum is what produces progesterone, estrogens, relaxin, and inhibin, and that occurs until it degenerates into whatever fibrous scar tissue, which is commonly referred to as corpus albicans. Now, as I said prior about all the super important things that the female reproductive system does, one of it is known as oogenesis. So this is just the formation of gametes in the ovaries. So this is like spermatogenesis in the males. Okay. Now spermatogenesis begins in the males at puberty. However, oogenesis begins in females before they're even born. Okay. This whole process starts way before this. Okay. And it occurs in essentially the same manner as spermatogenesis. Okay, goes meiosis and meiosis one and two. Okay, and eventually it makes germ cells. However, germ cells then have to go maturation, just like the sperm had to when they got their mobility and their ability to fertilize. So during early fetal development, primordial or primitive germ cells, they migrate from the umbilical vesicle to the ovaries. Now, their germ cells differentiate within the ovaries into oogoneas. These guys are diploid. Okay, they're diploid stem cells, and they divide mitotically to produce millions of germ cells. Now, even before birth, most of these germ cells degenerate in a process known as atresia. So a few of them, however, will develop into larger eggs. Those are the primary oocytes. These then enter prophase of meiosis one during fetal development, but they don't actually complete that phase until after puberty. So during this arrested stage of development, each primary oocyte is surrounded by a single layer of flat follicular cells, and the entire structure is what is known as the primordial ovarian follicle. The ovarian cortex surrounding this consists of those collagen fibers, those fibroblast-like stromal cells, and at birth, there's around 2 million primary oocytes that actually end up remaining in each ovary. So of those 2 million, about 40,000 are actually going to remain present at puberty. And eventually, when a woman is mature, it'll be about 400. Okay, so there is this shift over time of the reproductive lifetime of these primary oocytes. Now, each month after puberty, all the way until menopause, gonatrophins secreted by the anterior pituitary further stimulate the development of several primordial ovarian follicles, although only one will typically reach the maturity needed for ovulation. So a few primordial ovarian follicles start to grow. They develop in the primary ovarian follicles, but each primary ovarian follicle consists of a primary oocyte that is surrounded in a later stage of development by several layers of cuboidal and flow columnar cells called gangliosa cells. The outermost of those granulosa cells rest on what is known as a basement membrane. And as the primary ovarian follicle grows, it forms this clear glycoprotein layer called the zona pellucida. And this occurs between the primary and granulosa cells. Now, in addition to these stromal cells also surround the basement membrane, and they also help to then furthermore form an organized layer that's known as the theca follicle. Now, when continuing maturing occurs, okay, a primary ovarian follicle develops into a secondary follicle. So in a secondary one, the theca differentiates into two layers. Theca interna, which is highly vascularized. It's just the internal layer of cuboidal secretory cells, and it secretes androgens. And then it can turn into the theca externa, which is an outer layer of stromal cells and collagen fibers. Now, in addition, granulosa cells can begin to secrete follicular fluid, which builds up in a cavity called the atrium of the center of the secondary ovarian follicle. And the innermost layer of these granulosa cells becomes firmly attached to the zona pellucida, and is now what we know as a corona radii. Now, the second ovarian follicle is or does eventually become larger. Okay, it turns into a tertiary ovarian follicle. While in this ovarian follicle, and just before ovulation occurs, this diploid primary oocyte, it finally completes meiosis one, and it produces two haploid cells, actually of unequal size, each with 23 chromosomes. Okay, the smaller cell produced by meiosis one is called the first polar body, and essentially a packet of discarded nuclear material. The larger cell is known as the secondary oocyte, and it receives most of the cytoplasm, hence the change in sizes. Once a secondary oocyte is formed, it begins meiosis two, but then stops in metaphase. So it's kind of this pattern of stop, pause, okay? And the tertiary follicle soon ruptures and releases its secondary oocyte, and this becomes the process of ovulation. Okay, so at ovulation, the secondary oocyte is expelled into the pelvic cavity together with the first polar body and corona radio. So normally these cells are swept into the uterine tube, and if fertilization does not occur, the cells degenerate. But if sperm's present in the uterine tube and one penetrates the secondary oocyte, however, meiosis 2 resumes, and the secondary oocyte splits into two haploid cells. Again, it follows an unequal size pattern, where the larger cell is the ovum or mature egg, and the smaller one is the second polar body, just that nuclei waste. Okay, the nuclei of the sperm and the ovum unite, that forms a diploid zygote. So if the first polar body undergoes another division to produce two polar bodies, then the primary oocyte ultimately actually gives rise to three haploid polar bodies, which all degenerate, and only a single haploid ovum. So one primary oocyte gives rise to a single gamete, but by contrast, if you remember in males, one primary spermatocyte produced four gametes. Okay, so there's a variation in these similar processes on exactly what occurs at the end. And furthermore, in this process, how much time in between the pausing of phases as meiosis and meiosis 2 is occurring. To the male reproductive system, the female comes with a lot of tubes and a lot of ducts. One of the main ones are the uterine tubes. Okay, so the females have two of these tubes. They're also called fallopian tubes, sometimes oviducts. Okay, and they extend laterally from the uterus. The tubes are decent size. They're about 4 inches long, and they lie between the folds of the broad ligaments of the uterus itself. Okay, they provide a route for sperm to reach an ovum and transport secondary oocytes from fertilized ova from the ovaries to the uterus. The funnel-shaped portion of each tube is called the infundibulum and is close to the ovary, but it's open to the pelvic cavity actually. Now, it ends in a fringe of these finger-like projections called fimbria, and one of which is attached to the lateral end of the ovary. Now, from the infundibulum, the uterine tube extends medially and eventually inferiorly and attaches to the superior lateral angle of the uterus. The ampulla of the uterine tube is the widest, longest portion, making up about 2/3 of its entire length. Okay, now the isthmus of the uterine tube is the more short side, and it's a more thick-walled portion that is there where it joins the uterus. Now, histologically, the uterine tubes are composed of three layers: mucosa, muscular layer, and serosa. Mucosa consists of epithelium and lamina propria, or areolar connective tissue. The epithelium contains ciliated simple columnar cells, which function as a ciliary conveyor belt, so helping kind of keep things moving along. Okay, specifically a fertilized ovum. And they also consist of non-ciliated cells. Those are called PEG cells. Okay, they have microvilli actually, and they secrete a fluid that provides nutrition for the ovums. Now, the middle layer, the muscular layer, is composed of an inner thick circular ring of smooth muscle and an outer thin region of longitudinal smooth muscle. So, two different types of muscles. Peristaltic contractions of the muscular layer and the ciliary action of the mucosa is what helps then to move the outside of the fertilized ovum toward the uterus. And lastly, the outer layer of the uterine tubes is the serous membrane. And it's there formed by the visceral petroneum, which, if you'd expect, it's kind of assisting with protection, kind of keeping it all together. So after ovulation, local currents are produced by the movements of the femora, which surround the surface of the ovary just before the ovulation commences. These currents sweep the ovulated secondary oocyte from the peritoneal cavity into the uterine. Now, sperm usually encounters and fertilizes a secondary oocyte in the ampulla of the uterine tubes. Although fertilization in the peritoneal cavity is not common, it can occur. Okay, and fertilization can occur up to about 24 hours after ovulation. So, some hours after fertilization, the nuclear material of a haploid ovum and a sperm are going to unite, and the diploid fertilized ovum is now the zygote. And that's when it begins to undergo cell divisions, and it's all occurring while it's actually moving toward the uterus. Okay, and where it actually ends up arriving about 6 to 7 days after ovulation. And of course, if this is unfertilized, it just disintegrates. Okay, now, as we said, it makes its way to the uterus. So the uterus is also commonly known as the womb. Okay, and it serves as part of the pathway for sperm that's deposited in the vagina to reach the uterine tubes. And it's also the site of implantation of the fertilized ovum and development of the fetus during pregnancy as well as labor. During reproductive cycles, when implantation does not occur, the uterus is the source of menstrual flow. Looking at the uterus, it's situated between the urinary bladder and the rectum, and it's the size and shape of about a pear or an apple. Okay, and the uterus is actually different sizes, kind of among others, and age groups as well. So the uterus is larger in females who've recently been pregnant, but actually smaller when sex hormones are low. So that's some common features of women after they hit menopause. Okay, so various impacts of hormones, just environmental exposure, can impact the uterus's makeup, the anatomy of it. Now, the uterus has subdivisions, anatomical subdivisions. One, a dome-shaped portion that's superior to the uterine tubes called the fundus, a tapering central portion called the body, and an inferior narrow portion that's called the cervix that opens into the vagina. Between the body of the uterus and the cervix is the isthmus. So that's just a constricted region. The interior of the body of the uterus is called the uterine cavity, and the interior of the cervix is called the cervical canal. The cervical canal opens into the uterine cavity at the internal and into the vagina at the external. So normally the body of the uterus projects interiorly and superiorly over the urinary bladder. That's a position known as an anteflexum. The cervix projects inferiorly and posteriorly and enters the anterior wall of the vagina at nearly a right angle. So several ligaments that are either extensions of the parietal peritoneum or fibromuscular cords are what's there to help maintain that entire position of the uterus. Paired broad ligaments are double folds of peritoneum, and that also assists with attaching the uterus to either side of the pelvic cavity. Paired altersacral ligaments, they're also peritoneal extensions in a sense. These guys lie on either side of the rectum and connect the uterus to the sacrum as well. So the cardinal ligaments are located inferior to the base of the broad ligaments and extend from the pelvic wall to the cervix and the vagina. So what you kind of need to imagine is that it's kind of like it has a bunch of support beams that are holding it in the place that it needs to be held, which kind of makes sense because you're going to have some precious cargo in there following fertilization. You need to make sure it's steady and that it's protected as much as possible. Okay, so we have a lot of these connecting ligaments that are there to kind of help to hold it in place. Physiologically, the uterus consists of three layers. Okay, the perimetrium, the myometrium, and the endometrium. Now, the outer layer is the perimetrium, okay, or serosa, and it's part of the visceral peritoneum, and it's composed of simple squamous epithelium and areolar connective tissue. Laterally, it becomes the broad ligament. Interiorly, the peritoneum reflects off the uterus and it covers the urinary bladder, and that forms a shallow pouch known as the vescotarian pouch. Posteriorly, it covers the rectum and it forms a deep pouch between the uterus and the rectum, the rectoarian pouch, or pouch of Douglas, which is actually the most inferior point in the entire pelvic cavity. The middle layer of the uterus is the muscular layer, called the myometrium. And this consists again of three layers of smooth muscle fibers that are thickest in the fundus and thinnest in the cervix. The thicker middle layer is circular. The inner and outer layers are longitudinal or oblique. Now, during labor and childbirth, coordinated contractions of this myometrium is response to oxytocin from posterior pituitary, and that's actually what helps to contract and expel the fetus from the uterus. Then, lastly, the inner layer of the uterus is the mucosa. So this is highly vascularized. It's actually referred to as endometrium, and it itself has these three sublayers. The compact layer of the endometrium, this is the functional layer, and the basal layer of the endometrium. The compact layer is the most superficial layer and is composed of ciliated simple columnar epithelium and non-ciliated secretory cells, and it makes a very thick endometrial stroma. Okay, and it's composed of areolar connective tissue, portions of simple tubular uterine glands, all which develop as invaginations of the epithelium and extend through the other layers of the endometrium, almost to the myometrium. The functional layer consists of spongy endometrial stroma, okay, or lamina propria, and composed of the areolar connective tissue as well. So this one is rich in ground substance and includes most of the length of the uterine glands. The functional layer and compact layer are sloughed off during menstruation, and this is all in a response to declining levels of progesterone from the ovaries. The basal layer is also composed of endometrial stroma. Okay, that is more highly cellular and includes terminal ends of uterine glands. The basal layer is permanent and it contains stem cells that give rise to new functional compact layers after each menstruation cycle. Branches of the internal iliac artery within the uterus, because everything needs blood supply, right? Especially something like this. They're known as uterine arteries, and they give off branches called arcuate arteries. And these are arranged in circular fashion in the myometrium, and these arteries branch into radial arteries that penetrate deeply into the myometrium. So just before the branches enter the endometrium, they divide into two kinds of atrioles. Straight atrioles, which supply the basal layer of the materials needed to regenerate and compact and all the functional layers needed to do what they need to do. Blood leaving the uterus is then drained after it does its job, and that's drained by the uterine veins into the internal iliac veins. This extensive blood supply of the uterus is essential for obvious reasons because it's there to help to support the growth of new compact functional tissues, and menstruation, implantation of fertilized ovums, and of course the development of placenta. So as you'd expect, this is quite important and it's quite regulated. Now the secretory cells of the mucosa, okay, of the cervix, they produce a specific secretion. It's called the cervical mucus. This is just a mixture of water, glycoproteins, lipids, enzymes, and inorganic salts. And so during their reproductive years, females actually secrete about 60 milliliters of this just per day. Cervical mucus is more hospitable to sperm at or near the time of ovulation because it's less viscous, more alkaline, which is why they say tracking cycles, figuring out the ovulation is going to significantly increase your chance of being pregnant. Okay. Now, cervical mucus supplements energy needs of sperm as well. And both the cervix and cervical mucus actually protect it from the phagocytes. This is why it's so important. This is why it helps to increase the chance of pregnancy. It also may play a role in capacitation, which is just a series of functional changes that sperm undergo in female genitalia tract before they're actually able to fertilize. And capacitation causes like the tail of the sperm to beat more vigorously, kind of think of it like a rudder in a boat. And it prepares the plasma membrane of the sperm to fuse with the oocyte's plasma membrane, so making it more, don't want to say flexible, but fusible. So, the vagina is a tubular, long fibromuscular canal. It's lined with a mucous membrane that extends from the exterior of the body of the uterine cervix. It is the reciprocal for the penis during sexual intercourse, and it's the outlet of the menstrual flow, the passageway of the childbirth. Now, situated between the urinary bladder and the rectum, the vagina is directed superiorly and posterior, where it attaches to the uterus. A recess called the fornix surrounds the vaginal compartment and attachment to the cervix. When properly inserted, contraceptive diaphragm rests in that fornix where it's held in place as it covers the cervix. The mucosa of the vagina is continuous with that of the uterus. And histologically, it consists of non-keratinized stratified squamous epithelium and areolar connective tissue. Okay, and it lies in a series of transverse folds that are actually called the vaginal rugae. Dendritic cells in the mucosa are antigen-presenting cells. So unfortunately, they also participate in transmission of viruses. So, for example, HIV. Okay, female during intercourse with an infected male, unfortunately, can help with participating in the transmission of the virus. Mucosa of the vagina contains large stores of glycogen, the decomposition of which produces the organic acids, and the resulting acidic environment retards the microbial growth, but it can also become harmful to the sperm. Okay, so while it has its own defense mechanisms, it's kind of the double-edged sword. Okay. Alkaline components of semen, mainly from seminal glands, they are the ones that raise the pH of fluid in the vagina, and it's kind of like a combatant method of increasing viability of the sperm. The muscular layer of the vagina is composed of an inner circular layer and an outer longitudinal layer of smooth muscle that can stretch considerably. This accommodates for the penis during intercourse and the child during birth. The adventitia is the superficial layer of the vagina, and it consists of areolar connective tissue, and that's anchors the vagina to the adjacent organs. That's like the urethra, the bladder, the rectum, the anal canal. Kind of keeps it all in the location and position it needs to be. A thin fold of vascularized mucous membrane, which is called the hymen, forms a border around it and partially closes the inferior end of the vaginal opening to the exterior, the vaginal orifice. And it is a variable size in shape and sometimes actually may not even be there. Okay, so it's not a one size fits all. And sometimes it's completely covers the orifice, and that becomes a condition known as the infrared hymen. So surgery has to be done, and eventually it opens it, which allows the discharge of menstruation flow. So the vulva is all the external genitalities of the female. Okay, so it has several components. Now, anterior to the vagina and the urethra openings is the mons pubis. So it's an elevation of adipose tissue that's covered by the skin, the pubic hair, and that's what helps cushion the pubic symphysis. Now, from this, two longitudinal folds or skins, the labia majora, extend inferiorly and posteriorly. The singular term is labienus. Okay. The These are covered with pubic hair, adipose tissue, sebaceous glands, pseudorifice glands, and they are homologous basically to the scrotum in a sense in the male side of things. Medial to these guys are two smaller folds. Okay, they're called the labia minora. Now, this singular term again is labia minus. Unlike these guys, the majora, the minora are devoid of pubic hair, okay, or fat, but they do have those subdurifice glands. But they also contain many, much more than the prior, subcaseous glands, and that helps to kind of fight against substances as they produce antimicrobial substances and can even assist further by providing lubrication during intercourse. The clitoris is a small cylindrical mass that's composed of two small erectile bodies, the corpora cavernosa and numerous nerves with blood vessels. Okay, the clitoris is located at the anterior junction of the labia minora. A layer of skin called the prepuce of the clitoris is formed at the point where the labia minora unite and covers the entire body of the clitoris. The exposed portion of it is known as the glans clitoris and it's basically homologous to the glans penis in the males. Okay. So, but like the male structure, clitoris is capable of enlargement on tactile stimulation and it has a role in sexual excitement in the female as well. The region between the labia minora is the vestibule. Now, within this are the hymen, okay, the vaginal orifice and the external urethral orifice, as well as openings of the ducts of several glands. The vaginal orifice is the opening of the vagina to the exterior, which occupies the greater portion of the vestibule and is bordered by the hymen. Anterior to the vaginal orifice and posterior to the clitoris is the external urethral orifice. This is the opening of the urethra to the exterior. Now, on either side of the external urethral orifice are the openings of the ducts of the periurethral glands, also known as Skene's glands. These mucus secreting glands are embedded in the wall of the urethra, and they are basically homologous to the prostate on the male side. Now, on either side of the vaginal orifice itself are the greater vestibular glands. Okay, these are opened by ducts falling into a groove between the hymen and the labia minora. They produce small quantity of mucus during arousal, intercourse. It adds to cervical mucus and it does provide some lubrication. The greater vestibular glands are homologous to the bulbourethrial glands in the males. Okay, and there's several lesser vestibular glands that help to secrete mucus during sex and intercourse as well, which also open into the vestibule. Now, the ball of the vestibule, these consist of two elongated masses of erectile tissue just deep to the labia on either side of the vaginal orifice. The bulb of this becomes engorged with blood during sexual arousal, narrowing the vaginal orifice and placing pressure on the penis during intercourse. So the bulb of the vestibule is pretty much similar to the corpus spongiosum and the bulb of the penis in males having a similar response. The perineum is the diamond-shaped area medial to the thighs and the buttocks of both males and females. Because we both have this. And it contains the external genitalia, the anus. The perineum is bounded interiorly by the pubic symphysis, laterally by the ischial tuberosity, and posteriorly by the coats. A transverse line drawn between these divides the perineum into an anterior urogenital triangle, and this contains the external genitalia and our posterior anal triangle that contains the anus. The mammary glands are part of the reproductive system. Okay. Specifically, each breast is a hemispheric projection of variable size, okay, anterior to the pectoralis major and serratus anterior muscles, and attached by a layer of fascia composed of dense, irregular connective tissue. And each breast has one pigmented production, okay, the nipple. This has a series of closely spaced openings of ducts called the lactiferous ducts. This is where milk emerges during lactation. The circular pigmented area of the skin surrounding the nipple is called the areola, and it appears rough because it contains modified subcutaneous glands, strands of connective tissue called the suspensory ligaments of the breast run between the skin and the fascia, and that's kind of what helps to support the breast. These ligaments do become looser with age or with excessive strain, such as long-term jogging, high-impact aerobics, where you find that the tear of it leads to the need of more supportive bras, such as sports bras and sports. Now, within each breast is a mammary gland. Okay, so they're not really interchangeably, whereas breast is, you're talking about the whole thing. Okay, whereas mammary gland is this pseudoaphorous gland that actually then produces the milk. Okay, a mammary gland consists of several lobes, okay, or compartments that are separated by adipose tissue, fat tissue. And in each lobe are several smaller compartments that are called lobules. These are like kind of grape-like clusters They have milk secreting glands. Okay, they're called granular alveoli, and they're embedded in connective tissue within this whole system. Now, contraction of myoepithelial cells that surround the glandular alveoli is what actually helps to propel milk towards nipples. And when milk is being produced, it passes from the alveoli into a series of secondary tubules and then into the mammary ducts. Near the nipple, the mammary ducts expand slightly to form sinuses called the lactiferous sinus, and that's where some milk may be stored before draining into a duct. Now, each lactiferous duct that typically carries milk from one of the lobes to the exterior, so each is doing their part. Now, the functions entirely of the mammary gland is it assists with synthesis, secretion, and injection of milk. Okay, all termed lactation. Okay, everyone is associated with pregnancy and childbirth. Now, milk production is stimulated largely by hormone prolactin, okay, specifically from the anterior pituitary with contributions of progesterone and estrogens. And the ejection of milk is actually stimulated by oxytocin, and that's from the posterior pituitary. And a lot of times it's in response to the sucking of an infant on the mother's nipple, also known as suckling. So during the reproductive years, non-pregnant females normally go through cyclical changes in their ovaries and their uterus, and each cycle takes about a month. And it involves both the oogenesis and preparation of the uterus to receive a fertilized ovum. And hormones secreted by the hypothalamus, the anterior pituitary, and the ovaries control these main events. The ovarian cycle is a series of events in the ovaries and occurs during and after the maturation of an oocyte. The uterine or menstrual cycle is a concurrent series of changes in the endometrium of the uterus to prepare it for the arrival of a fertilized ovum that will develop there until birth. And if fertilization does not occur, ovarian hormones wane, which cause the functional layer of the endometrium to slough off. Okay. The general term female reproductive cycle encompasses the ovarian and the uterine cycles, everything together, the hormonal changes as well that regulate them, okay, and all the cyclical changes within the breast and cervix. Now, hormonal regulation has a key player with gonadotropin releasing hormone, GnRH. This is secreted by the hypothalamus and it controls the ovarian and uterine cycles. And it stimulates the release of the follicle stimulating hormone, or FSH, as well as the luteinizing hormone, LH, from the anterior pituitary. FSH initiates follicular growth, while LH stimulates further development of the ovarian follicles. Now, in addition, both stimulate the ovarian follicles to secrete estrogens, bringing in hormones, right? And LH stimulates the theca cells of a developing follicle to produce androgens. Now, under the influence of SH, the androgens are taken up by the granulosa cells of the follicle, and those then get converted into estrogens. At mid-cycle, LH triggers ovulation and then promotes the formation of the corpus luteum, and that's the reason for the name luteinizing hormone. Stimulated by the LH, the corpus luteum then goes forth and it produces and secretes estrogens, progesterone, laxin, and inhibin. At least 6 different estrogens have been isolated from the plasma of human females, but only 3 of them are present in significant quantities. That's beta estradiol, estrone, and estriol. And a non-pregnant woman, the most abundant estrogen is beta estradiol, which is synthesized by the cholesterol in the ovaries. Now, these guys, the ones that are secreted by the ovarian follicles, they have specific functions. Okay, these estrogens promote the development and maintenance of female reproductive structures, secondary sex characteristics, as well as their breast. The secondary sex ones, that's like distribution of adipose tissue, going to the breast, the abdomen, the hip growth, voice pitch, the broadening of the pelvis, pattern of hair growth. It helps to increase protein anabolism, which includes the building of strong bones. So in this regard, estrogens are actually synergistic with growth hormones. Helps with lowering blood cholesterol level, which is probably the reason that women under age of 50 have a much lower risk of coronary heart disease than do men at the same age. Every month after menstruation occurs, estrogens stimulate the proliferation of the basal layer that then forms a new functional layer that replaces one that has just sloughed off. Moderate levels in the blood inhibit both the release of GnRH by the hypothalamus and secretion of LH and FSH by the anterior pituitary. Progesterone, secreted mainly by cells of the corpus luteum, cooperates with the estrogens to prepare and maintain the endometrium for implantation of a fertilized ovum and to prepare the mammary glands for milk secretion. So higher levels of progesterone will also go forth and inhibit GnRH and LH secretion. The small quantity of relaxin that's produced by the corpus luteum during each monthly cycle, that relaxes the uterus, and it's done by inhibiting contractions of the myometrium. So presumably implantation of a fertilized ovum occurs more readily in a quiet uterus. During pregnancy, the placenta produces much more relaxin, and it continues to relax uterine smooth muscle. And at the end of pregnancy, relaxin increases the flexibility of the pubic symphysis and helps to dilate the uterine cervix, and both of which to go forth and ease the delivery of the baby for the woman. Another secretion is inhibin. Okay, it's also secreted by granulosa cells. This is it helps with ovulation. It's solely because it has an interaction with FSH and LH, so it's another form of kind of hormone feedback control. So phases of the female reproductive cycle. The duration of this whole cycle, it's usually 24 to 36 days. For this, we're just going to go 28 days. Okay, and it's existing in 4 phases: the menstrual phase, the pre-ovulatory phase, ovulation, post-ovulatory phase. Now, during the menstrual phase, also called menstruation or medes, I'm going to go menstruation. It lasts for about the first 5 days of the cycle. Okay, and during this time, under the influence of FSH, several primordial ovarian follicles develop into primary ovarian follicles and then into secondary ovarian follicles. And this whole developmental process that takes possibly several months to occur allows an ovarian follicle that begins to develop at the beginning of a particular menstruation cycle to reach maturity in several cycles. Okay. And now in the uterus, menstrual flow from the uterus, that's about 50 to 150 milliliters of blood. It's not just blood, it's tissue fluid, it's mucus, it's epithelial cells that shed from the endometrium. So that's why it's considered just flow instead of blood. This discharge occurs because the declining levels of progesterone and estrogen stimulate the release of prostaglandins and cause the uterine spiral arterioles to constrict. So as a result, the cells they supply become oxygen deprived, they start to die, and eventually the entire functional compact layers slough off. Okay, this thins the endometrium, and because only the basal layer remains, the menstrual flow passes from the uterine cavity through the cervix and the vagina to the exterior. In the pre-ovulatory phase, this is the time between the end of menstruation and ovulation. Okay, and it's more variable in length than all these other phases. It's because there's a lot of differences in environment and hormones and age, all those different things you have to take into account. Okay, but so what happens in the ovaries during this phase is that some of the secondary ovarian follicles that are in the ovaries, they begin to secrete the estrogens and inhibin. And by about day 6, a single secondary ovarian follicle in one of the 2 ovaries has outgrown all the others to become the dominant ovarian follicle. Estrogen and inhibin secreted by the dominant ovarian follicle decreases secretion of FSH, which causes less well-deprived ovarian follicles to stop growing, and then they degenerate. Fraternal or non-identical twins, sometimes even triplets, result when 2 or 3 secondary ovarian follicles become codominant, And so later they're ovulated and fertilized around the same time because they weren't degenerated. Normally, the one dominant secondary ovarian follicle becomes a tertiary ovarian follicle, okay, which then continues to enlarge until it's more than a certain size, okay, and then it's ready for ovulation. The ovarian follicle forms what looks like a blister-like bulge due to swelling antrum on the surface of the ovary. And during the final maturation process, the tertiary ovarian follicle continues to increase the further production of estrogens. Now, with reference to ovarian cycle, the menstrual and proovulatory phases together, these are termed the follicular phase. Because ovarian follicles are growing and developing and changing and being degenerated. Now, all these changes in estrogens, okay, estrogens are being liberated into the blood, okay, all by these growing ovarian follicles. It stimulates the repair of the endometrium, or the cells of the basal layer. They start to undergo mitosis, produce new functional compacted layers. As the endometrium thickness, the short, straight endometrial glands develop, and they rolls coil and lengthen as they penetrate the functional layer, and the thickness of the endometrium starts to increase, starts to double over time. Now, looking at the uterine cycle, the pre-ovulatory phase is termed the proliferative phase because of this. The endometrium is proliferating, it's healing, it's growing. So, ovulation, all right, it's the rupture of the tertiary ovarian follicle and the release of the secondary oocyte into the pelvic cavity. This is around day 14 in that 28-day cycle. During ovulation, secondary oocyte remains surrounded by its sona fluid and corona radiata. The high levels of estrogens during that last part of the preovulatory phase exert this positive feedback effect on cells that secrete the LH and the GnRH and then causes ovulation. Okay, and so how it does this is there's a high concentration of estrogens that goes and stimulates the release of GnRH from the hypothalamus, and it also directly stimulates gonadotrophins in the anterior pituitary to secrete LH. So GnRH promotes the release of FSH and additional LH by the anterior pituitary. Now, LH causes rupture of the tertiary ovarian follicle and expulsion of the secondary oocyte. It's at the peak of the LH surge. The ovulated oocyte and its corona radiata cells are usually swept into the uterine tube. Now, from time to time, an oocyte is lost into the pelvic cavity where it later disintegrates. The small amount of blood that sometimes leaks into the pelvic cavity from the ruptured follicle, it's caused pain. It's known as the Mittelschmeier's that occurs at the time of ovulation. Now, getting into that post-ovulatory phase, this is the phase of the reproductive cycle that is between ovulation and the onset of the next cycle. Okay, and in duration, it lasts for about 14 days in that 28th cycle, so from day 15 to 28. Now, after ovulation, the tertiary ovarian follicle collapses, and the basement membrane between the granulosa cells and the thick antenna breaks down. And once a blood clot forms from minor bleeding of the ruptured ovarian follicle, the follicle becomes the corpus hemorrhagicum. And then theca interna cells mix with the granulosa cells as they all become transformed into the corpus luteum cells and under the influence of LH completely. Okay, now stimulated by LH, corpus luteum secretes progesterone, estrogens, relaxin, and inhibin. And the luteal cells also go forth and absorb the blood clot that was made. Now, later events in an ovary that are ovulated in an oocyte depend on whether it is successfully fertilized. And if it's not fertilized, the corpus luteum has a lifespan of only 2 weeks. Then its secretory activity declines and it degenerates into a corpus albicans. And as the levels of progesterone, estrogen, and inhibin decrease, release of GnRH, FSH, and LH rise due to the loss of negative feedback suppression by ovarian hormones. Then follicular growth resumes and a new ovarian cycle begins. If the secondary oocyte is fertilized and it begins to divide, the corpus luteum persists past its normal 2-week lifespan, and it is rescued from degeneration by human chorionic gonadotropin. This hormone is produced by the colon of the embryo, and it's about 8 days after fertilization that it's made. And so, like LH, this hormone stimulates the secretory activity of the corpus luteum, and the presence of it in maternal blood or maternal urine is an indicator of pregnancy. And it's actually what sometimes it's used for at-home pregnancy tests. Now, events in the uterus during this time, we have hormones just soaring, right, going all over. So estrogens, progesterones that are produced by the corpus luteum, they promote the growth and the coiling of these endometrial glands, as well as their vascularization of the superficial endometrium and the thickening of it. So because of the secretory activity of the endometrial glands, which begin to secrete glycogen, the period is often called the secretory phase, the uterine cycle. These preparatory changes, they go and peak about 1 week after ovulation. So at the time a fertilized ovum might arrive in the uterus, if fertilization does not occur, the levels of progesterone and estrogens decline due to degeneration of the corpus luteum, and withdrawal of progesterone and estrogens cause the menstruation to go forth and occur. So during heterosexual intercourse, also called copulation or coitus, the erect penis is inserted into the vagina. The similar sequence of physiological and emotional changes experienced by both males and females occurring before, during, and after intercourse. That's the human sexual response. Now, the sexual response is designated into 4 phases: excitement, plateau, orgasm, and resolution. During the excitement phase, there is vasocongestion, or the engorgement with blood of genitalia tissue, resulting in the erection of the penis in men and the erection of the clitoris and swelling of the labia and vagina in women. In addition, the vasocongestion causes the breasts to swell, the nipples to become erect as well. The excitement phase is also associated with an increase in the secretion of fluid that lubricates the walls of the vagina. And when the connective tissue of the vagina becomes engorged with blood, lubricating fluid oozes from the capillaries and seeps through the epithelial lining. This process is known as transudation. Glands within the cervical mucosa and the greater vestibular glands contribute a small quantity of lubricating mucus, all in preparation of incoming sex. Now, without satisfactory lubrication, sexual intercourse is difficult and painful for both partners, and it can inhibit orgasms. Other changes that occur during the excitement phase include increased heart rate, blood pressure, skeletal muscle tone throughout the body, and sometimes hyperventilation. Direct physical contact, this is hugging, kissing, touching, all that, especially of the genitalia, like penis, clitoris, nipples of the breast, and sometimes even earlobes, is a potent initiator of excitement. However, anticipation of fear, fearful memories, any visual, olfactory sensations that may be off putting can either enhance and sometimes diminish the likelihood of excitement to even occur. Now, the changes that begin during excitement are sustained at an intense level in the plateau phase, which may last for a few seconds or for many minutes. During this phase, many females and some males display a sex flush. This is a rash-like redness of the face and chest, and it's all done by the vasodilation of blood vessels in those specific regions of the body. The head of the penis increases in diameter, and the testes swell. And late in the plateau phase, pronounced vasocongestion of the lower third of the vagina swells. The tissue then narrows the opening, and because of this response, the vagina grips the penis more firmly. Generally, the briefest phase is the orgasm, also known as climax. This is at the point when sex is experienced several rhythmic muscular contractions. This is accompanied by intense pleasurable sensations and a further increase in blood pressure, heart rate, respiratory rate. The sex flush is also most prominent at this time. In males, contraction of smooth muscle on the walls of the epididymis, vas deferens, and ejaculatory ducts, as well as secretion of fluid by the accessory male genitalia glands, cause semen to move into the urethra, or emission. Then rhythmic contractions of the skeletal muscle at the base of the penis propel semen out of the penis, or ejaculation, and in males, orgasms usually accompany ejaculation. In women, if effective sexual stimulation continues, orgasm may occur, and it's also associated with 3 to 12 rhythmic contractions of the skeletal muscles which underlie the vulva. Reception of the ejaculate provides little stimulus for a female, especially if she is not ready at the plateau phase. This is why a female partner does not automatically experience orgasm simultaneously with her partner. Now, in both males and females, orgasm is a total body response that it can produce mild sensations on some occasions and more intense on others, sometimes even explosive. Whereas females may experience 2 or more orgasms in a rapid succession, males enter a refractory period, that is a recovery time, during which a second ejaculation orgasm is physiologically impossible. In some males, the refractory period lasts only a few minutes. In others, it lasts for several hours. A female does not have to experience an orgasm for fertilization to occur, compared to its male counterpart, where it does. Now, in the final phase, resolution, which it begins with a sense of profound relaxation, genital tissues, heart rate, blood pressure, breathing, muscle tone returning to the unaroused state, just relaxation occurring. If sexual excitement has been intense but orgasms has not occurred, resolution takes place at a more slow manner than it would have on the flip side. So the 4 phases of the human sexual response, they're not always clearly separated from one another. And it can considerably vary among different person and even in the same person at different times. So it's not a one size fits all. Now, while we are designed, okay, to proliferate, uh, to have this type of sexual reaction to give off to our offspring, sometimes it needs to be controlled. Okay, so that's when we get into birth control, contraception, the ability to restrict the number of children by various methods. Okay. Methods of controlling fertility and preventing conception. No single ideal method of birth control exists. The only method of preventing pregnancy that's 100% reliable is complete abstinence. Now, the avoidance of sexual intercourse may not be as easy for others. So there's several other methods. Okay, each has its advantages, each has its disadvantages. These include surgical sterilization, hormonal methods, intrauterine devices, spermicides, barrier methods, and periodic abstinence. Now, although it's not a form of birth control, we also will discuss abortion in a bit, or the premature expulsion of products of conception from the uterus. But looking at some more, I guess you could say, controlled side of things, surgical sterilization is a procedure that's heavily used, okay, and it renders an individual incapable of further reproduction. The principal method for sterilization of male is just a simple vasectomy. That's where a portion of each ductus deferens, vas deferens, is removed. And this is in order to gain access to the ductus deferens. An incision is made with a scalpel, or a puncture is made with special forceps, and the ducts are located and cut. Each is tied in 2 places with stitches, and the portion between the ties is removed. So although sperm production is continuing in the testes, sperm can no longer reach the exterior. Okay, it's like it's hit a block in the road. Because of this, the sperm then become destroyed. They degenerate by phagocytosis. Now, because the blood vessels are not cut, testosterone levels in the blood remain normal. All right, so there's no impact of hormones. Okay, so it has no effect on sexual desire or performance. And if done correctly, it's really close to 100% effective. All right, and one of the good things that makes this ideal is this is a reversible procedure. Okay, so that if it is done, decision is made to go back to be able to reproduce, this is easily reversed. Now, sterilization in females on the surgical side, it's most often achieved by performing a tubal ligation.
Now, this is when both uterine tubes are tied and closed and then cut. Now, this can be achieved in a few different ways. Clips or clamps can be placed on the uterine tubes, and the tubes can be tied and cut out or cut out. And sometimes they are cauterized. That is, that they are destroyed by heat to stop the bleeding. In any case, the result is that the secondary oocyte cannot pass through the uterine tubules and sperm cannot reach the oocyte. So that's preventing, the fertilization from occurring. Now, non-incisional sterilization, there's tons of methods. Okay. This is including, alternative to the tubal ligation. It's like your micro insert coils that are made of polyester fibers or metals that become inserted via catheter to the vagina throughout the uterus or through the uterus and into each uterine tube. Now, over a 3-month period, insert stimulates tissue growth, so we get scar tissue in and around itself, and it blocks the uterine tubes. Now, as with this ligation, secondary oocyte cannot pass through the uterine tubes and sperm cannot reach the oocyte. But unlike tubular ligation, non-incisional sterilization does not require you to undergo general anesthesia, which is why it's commonly sought after. Now, one of the most common ones and one of the most, I guess you can say, oldest used is hormonal methods. Okay, so besides just completely going abstinent or having the surgical sterilization, hormonal methods are the next most effective means of birth control. And there's oral contraceptives, commonly referred to as the pill. Those contain hormones designed to prevent pregnancy. Some are called combined oral contraceptives. They contain both progestin, or hormone with action similar to progesterone, and estrogens. And now the primary action of these COCs is to inhibit ovulation by suppressing those gonadotropins FSH and LH. And the low levels of FSH and LH, that usually prevents the development of a dominant follicle in the ovary. So because of that, levels of estrogens do not rise. The mid-cycle LH surge then does not occur and ovulation does not take place. So even if ovulation does occur, as it sometimes does in some cases, COCs may also block implantation in another way. And it's how the uterus inhibits transportation of the ova and sperm and uterine tubes. So kind of preventing again from fertilization and implantation. Now, other parts that kind of play a role, progestins are known to thicken the cervical mucus. Okay, so because of this, it makes it more difficult for sperm to enter the uterus. Okay, and there's progestin only pills, which do exist. And among the non-contraceptive benefits of oral contraceptives and regulation of the length of menstrual cycle and decreased menstrual flow, the pill also provides protection against endometrial and ovarian cancers and actually reduces the risk of endometriosis. Now, oral contraceptions may not be advised for women with a history of blood clotting disorders because, and cerebral blood vessel damage, headaches, hypertension, liver, and a whole bunch of stuff. Okay, and it's because of the hormones that are involved in it, such as the progestins and the side effects they have outside of the plain mucosa involved. Now, there's other methods, okay, of hormonal methods. The combined pill. The combined pill contains both the progestin and estrogens. This one is taken about once a day for 3 weeks to prevent pregnancy. Okay, and it helps by regulating the menstrual cycle. Now, other things can help, such as skin patches, contraceptive skin patches. Again, a lot of this is hormonal, it's progestin and estrogens, but it's delivered in a skin patch from the outer arm. Vaginal contraceptive rings, which is just a donut-shaped ring that is placed into the vagina, and it contains estrogens, progesterones. The female inserts it herself, so again, it's ideal to not have to deal with the doctor. And it's just left in the vagina for a couple weeks, allowing the absorption of progesterones and estrogens. And of course, there are emergency contraceptions, so commonly known as the morning after pill. So that again is progestins and estrogens, or progestin alone, and it's again there to prevent the following unprotected sexual intercourse. Now, abortions, while commonly scrutinized, it's just a standard definition of it's just a premature expulsion of products of constant conception before 20 weeks. Now, while in the limelight right now, it's the induced intentional abortions that are focused on. Okay, but we do have spontaneous abortions. Those are commonly referred to as miscarriages, okay, to avoid the terminology of abortions. Okay, so there's two different types of abortions. Now, some of them are there mostly for medical reasons, okay, either for the baby or for the mom or for both, okay. These medication-based abortions is one version, but they're all done by a certain time point of the baby's development. So before 9 weeks, certain drugs can be taken. Okay. And of these certain drugs, mifepristone and misoprostol, they can either block the progesterone, which break down the endometrium, or stimulate uterine contractions, which then just expels the uterine contents. Okay. And there's surgical methods. Okay, again, there's different types of surgical methods, and it really depends on the baby's development. Vacuum aspiration is the most common because it's usually done early on in weeks, and it's just suction and removal of lining, placenta, embryo. Um, Dilation and evacuation, very similar, done 13, 16 weeks. It's enforcing the dilation by surgical means and removing the embryo fetus, placenta, and lining. Now, late stage, this is much more commonly used for uh, issues with the baby's health, the mother's health, or sometimes stillborns. Okay, and that is in the late stage, which is more for surgically opening um, the mother or inducing the labor to then pass the child. So, what happens if the deed gets done and we're starting to develop? Well, if you remember back from chapters ago, all right, somatic cells are diploids, right? So they contain 23 pairs of homologous chromosomes. That's a total of 46. So, of those chromosomes, there are 22 pairs of autosomes and one pair of sex chromosomes. Autosomes code for the overall form of the human body and for specific traits such as our eye color, our height, and others. Okay, and the two sex chromosomes, X and Y, go and determine the genetic sex of an individual. Now, in a genetic female, somatic cells contain the two X's. In a genetic male, it's an X and a Y. Okay, and so in gametes, all right, so sperms or eggs, these are haploid. Right, so there are only 23 total chromosomes, and of these chromosomes, there are 22 autosomes and one sex chromosome. So either an X or Y. So about half of the sperm produced by meiosis contain an X, and the other half contain a Y. In an egg, the sex chromosome is always an X, and that's because the mom is XX. Okay, so it's one way or another, it's getting an X. Now, genetic sex is established by at the moment of conception, okay, by the type of sperm that is entered. So it's either X bearing or Y bearing, okay, and that fertilizes the egg. And if it's an egg bearing sperm and fertilizes the egg, the embryo automatically becomes genetic female because it's XX. If it enters and it's a Y bearing, it automatically becomes a male, XY at the genetic sex. So the early embryo, when this is all occurring, the sperm has got into the egg, we have begun the implantation, fertilization, for so much. But the early embryo is biopotential, which means that it has the ability to form either male or female genitalia organs. And the first step in the development of the genitalia organs occurs in response to the genetic sex of the embryo. If the embryo is genetically male, testes develop. If the embryo is genetically female, ovaries develop. Once testes form in a male embryo, they begin to secrete androgens. All right, so masculinizing hormones. This causes a male genitalia tract and a male external genitalia to develop. Female embryos, which contain ovaries instead of testes, they do not produce testicular androgens. The lack of testicular androgens in a female embryo causes the female genitalia tract and female external genitalia to develop by default. So such a default pathway is ideal because both male and female embryos are exposed to high levels of estrogens and progesterones from the mother's placenta and ovaries during pregnancy. If female sex hormones played a role in sex differentiation, then all embryos would, whether genetically male or female, would develop female genitalia organs. Sex differentiation is the process by which genital organs develop along male or female lines. So, understanding the steps involved in sex definition, you have to first understand how internal genital organs are formed, and then we'll discover how the external genitalia are developed. So the gonads first, they develop from gonadal ridges. These arise from the growth of intermediate mesoderm. This is during the fifth week of development, about. Okay, the gonadal ridges appear as bulges just medial to the mesophores, okay, or intermediate kidneys. And adjacent to the gonadal ridges are the mesonephric ducts. These eventually develop in the structures of the genital system, in males specifically, and a second pair of ducts, the parasympathetic ducts, which develop lateral to the mesenteric ducts and eventually form structures of the genital system in females. Both sets of ducts empty into the urogenital sinus. An early embryo has the potential to follow either the male or the female pattern of development because it contains both sets of ducts and genital ridges that can either differentiate into testes or ovaries. All right, so we have the ability to do either. Now, cells of a male embryo have one X chromosome and one Y. The male pattern development is initiated by a master switch gene on the Y chromosome that's called SIR, SRY. This stands for sex determining region of the Y chromosome. So when this gene is expressed during development, its protein product causes primitive nurse cells to begin to differentiate into testes during the seventh week of development. The developing nerve cells secrete a hormone called Mullerian inhibiting substance, or MIS, which causes apoptosis of cells within that parasympathetic duct. So as a result, those cells don't contribute any functional structures to the male reproductive system. Now, stimulated by human clonal gonadotropin, or HCG, primitive interstitial endocrine cells in the testes begin to secrete the androgen testosterone during the eighth week. Testosterone then stimulates the development of the mesenteric duct on each side into the epididymis, ductus deferens, ejaculatory duct, and the seminal glands. The testes connect to the mesenteric duct through a series of tubules that eventually become the seminiferous tubules. Now, the prostate and the bulbal urethral glands are endodermal outgrowths of the urethra. Cells of a female embryo have two X chromosomes, so no Y chromosome. Because the SRY is absent, the gonadal ridges develop into ovaries. And because MIS is not produced, parasympathetic ducts grow. They're fine, they flourish. The distal ends of the parasympathetic ducts fuse to form the uterus and the vagina, and unfused proximal portions of the ducts become the uterine tubules. Now, the mesenteric ducts degenerate without contributing any functional structures to the female genitalia system because there's no testosterone. Now, the greater lesser vestibular glands, they develop from the endodermal outgrowths of the vestibule. Like the internal genitalia, the external genitalia of both male and female embryos, like the penis, scrotum, clitoris, labia, vaginal orifice, they remain undifferentiated until about the eighth week as well. So each, though, before differentiation, have similar external structures. External structures like the paired urethral or urogenital folds, which develop in the mesoderm and the colloquial region. An indentation between these urethral folds that is known as the urethral groove, which is the opening into the urogenital sinus. The genital tubercle, which is a rounded elevation just anterior to those urethral folds. And the labial scrotal swelling. Okay, that consists of paired elevated structures that are lateral to those urethral folds. Now, in male embryos, some testosterone is converted to a second androgen. This is called dihydrotestosterone, or DHT. This stimulates the development of the urethra, prostate, and external genitalia, the scrotum of the penis. Part of the genitalia tubercle elongates and develops into a penis, and fusion of the urethral folds forms the spongy urethra and leaves an opening to the exterior only at the distal end of the penis, the external urethral orifice as well. Now, the labioscrotal swellings develop into the scrotum, and in the absence of this DHT, the genitalia tubercle gives rise to the clitoris in female embryos. The urethral folds then remain open as the labia minora, and the labioscrotal swellings become the labia majora. The urethral growth becomes the vestibule, and after birth, androgen levels decline because of this HCG is no longer present to stimulate the secretion of testosterone, further helping with sexual orientation development. Any of these systems, age strongly impacts the genitally reproductive systems. And we actually have changes throughout our life, ups and downs as we go. So during our first decade of our life, the genitalia system is in a juvenile state. When we hit about age 10, hormone-directed changes start to occur in both sexes. You guys know that as puberty. Okay, the period when secondary sexual characteristics develop, and the potential for sexual reproduction is reached. Now, the onset of puberty is marked by the pulsive burst of LH and FSH secretion, each which is triggered by the pulse of GnRH. Now, most pulses occur during your sleep, which is why you hear growth spurt in your sleep, growth spurt at night. Now, as puberty advances, the hormone pulse occurs during the day as well as the night. So the pulses increase in frequency during about a 3 to 4 year period until the adult pattern is established. The stimuli that cause that GnRH pulse, it's pretty much still unclear. We don't really know, but the role of the hormone leptin is kind of starting to unfold. And just before puberty, leptin levels rise in proportion to the adipose tissue mass. Now, interestingly, leptin receptors are present in both the hypothalamus and anterior pituitary. And what they saw, if you had a mouse that would lack a functional leptin gene from birth, they are sterile. They remain in a prepubertal state. So leptin has this illicit secretion of gonadotropins that allows them become fertile, it's sexually mature. And leptin may signal the hypothalamus with the long-term energy storage, allowing it to provide an adequate energy for reproductive functions to begin. Now, in females, the genital cycle normally occurs once each month from menarche in the first minutes. This goes all the way to menopause, which is the permanent cessation of menses. So the female genitalia system has a time-limited span of fertility between menarche and menopause. So for the first 1 to 2 years after menarche, ovulation only occurs in about 10% of the cycles. Okay, and the lateral phase is short. So gradually, the percentage of ovulatory cycles increases, and the lateral phase reaches its normal duration of 14 days. Now, with age, fertility is going to decline. Between the ages of 40 and 50, the pool of remaining ovarian follicles becomes exhausted. So as a result, the ovaries become less responsive to hormonal stimulation, and the production of estrogen declines. And that's despite the secretion of FSH, and LH. Now, many women experience hot flashes or heavy sweating, and that's coinciding with the burst of the GnRH, okay, side effects of this increased secretion. Other symptoms of menopause, which you may have heard of, are headache, hair loss, muscle pains, vaginal dryness, insomnia, depression, weight gain, a whole lot of things that seem to be hormonal related. Okay. And by hormonal related, I'm talking about estrogens. Okay. It's occurring because there's a loss of estrogens, which leads as well to things like decline in bone mineral density after menopause, libido decreasing, sexual desire has gone down. It does not show a similar thing in their younger life or the previous lives. Interesting, it has some positive impacts. Okay, so actually some protective impacts against ovarian cancer, um, testicular cancer. So not all bad. Okay, now on the male side, um, we will start to lose our ability to complete spermogenesis. Okay, our sperm production will decrease at a staggering amount to 50 to 70% between ages 60 to 80. So while we still have some present, okay, it's not as efficient. So there's less chance for pregnancy to occur. Now, also along the side of that is enlargement of the prostate. So they enlarge is like 2 to 4 times its normal size, and it's usually those over the age of 60. That's known as benign prostatic hyperplasia. Now, decreases the size of prostatic urethra, and it's characterized by frequent urination, but sometimes this can lead to cancer, okay, prostate cancer. So we've talked about almost the development of every single system, okay? So now we're going to put it all together. All right. Then it's going to involve everything we just talked about. Okay. Male gamete sperm, female gamete secondary oocytes are, the testes making the sperm, the ovaries assisting with the secondary oocytes, and eventually comes together and sperm can be deposited in the female genitalia tract and find a secondary oocyte and be fertilized. Okay. And this whole process initiates pregnancy, okay, or the development of a healthy newborn baby. And as you'd expect, it's extremely tightly regulated. And it falls on a series of events, okay, fertilization, implantation, embryonic development, and fetal development. Now, if everything goes right, usually around 38 to 40 weeks after the mother's last menstrual period or the fertilization, the baby's born and it's healthy. Okay. Now, for fertilization, through our 8th week of development, this is known as the embryonic period. Okay, this is where the developing human is now becoming an embryo. After that 8 weeks period, we've now switched into what is known as the fetal period. Now, this begins at 9 weeks and continues all the way until birth. And during this time, the developing human, okay, goes from an embryo to a fetus. Now we go into prenatal development, the last phase of this process, these key time points. And this is the time from fertilization to birth and includes both embryonic fetal periods. And the prenatal development is divided into 3 different calendar months, each called trimesters. Okay, so during the first trimester, the most critical stage of development, all the major organ systems begin to form. We talked a lot about this in each chapter. Okay, and it's all because of this extensive, widespread activity. And it's also a period when the developing organism is most vulnerable to drugs, radiation, and microbes. The second trimester is characterized by the nearly complete development of an organ system. So by the end of this stage, the fetus assumes distinctively human features. Okay. The third trimester, the last one, represents a period of rapid fetal growth in which the weight of the fetus doubles. Okay, now during the early stages of this period, most of the organ systems basically become fully functional. Starting from the beginning and getting all the way through, the embryonic period extends from fertilization through the 8th week. Okay, and the first week of development is characterized by several significant events, and this is the fertilization, the cleavage of the zygote, the blastocyst formation, and the implantation. Okay, so during fertilization, genetic material from the haploid sperm and a haploid secondary oocyte merge into making a single diploid nucleus. Okay, and now of like the 200 million sperm that gets introduced into the vagina, fewer than 2 million, 1% will reach the cervix of the uterus, and only about 200 total will reach a secondary oocyte. And fertilization then will normally occur in the uterine tube, and it's usually within 12 to 24 hours after ovulation. Interesting fun fact, sperm can actually remain viable for about 48 hours after being deposited to the vagina, and that's although the secondary oocyte is viable for only about 24 hours after ovulation. So pregnancy is actually more likely to occur if intercourse takes place during a 3-day window, from 2 days before ovulation or to 1 day after ovulation. Now, sperm swim from the vagina into the cervical canal by this whip-like movement of their tails. Okay, and the passage of sperm through the rest of the uterus and into the uterine tube
results mainly from contractions of the walls of these organs. Prostaglandins and semen are believed to stimulate uterine motility. So at the time of intercourse, it aids the movement of sperm through the uterus and into the uterine tube. When sperm reaches the vicinity of the oocyte within minutes after ejaculation, they are not capable of fertilizing it until about 7-ish hours later. During this time in the female genitalia tract, mostly in the uterine tube, sperm undergo what is known as capacitation. This is just a series of functional changes that cause the sperm's tail to beat even more vigorously. It prepares its plasma membrane to be able to fuse with the oocyte's plasma membrane. And during this system, sperm are acted on by secretions in the female genitalia tract, and that results in the removal of cholesterol, glycoproteins, and proteins from the plasma membrane around the head of the sperm. So only capacitated sperm can be attracted by and respond to chemical factors, okay, produced by the surrounding cells of the ovulated oocyte. So after fertilization, rapid mitotic cell divisions of the zygote occur. It's called cleavage. It takes place. And the first division of the zygote begins about 24 hours after fertilization, and it's completed about 6 hours later. Each succeeding division takes slightly less time. So by the second day after fertilization, the second cleavage is completed, and there are 4 cells. By the end of the third day, there are 16 cells, and the progressively smaller cells produced by cleavage become blastomers. Successive cleavages eventually produce a solid sphere of cells that's called a merle, and the merle is still surrounded by the zona pellucida and is about the same size as the original zygote was. Now, by the end of the fourth day, the number of the cells in the merle increases as it continues to move through the uterine tube toward the uterine cavity. When the marrow enters the uterine cavity, it's usually around day 4 or 5, and glycogen-rich secretion from the glands of the endometrium of the uterus passes into the uterine cavity and enters the merula through the zona pellucida. The fluid, called uterine milk, along with other nutrients that are stored in the cytoplasm of blastomeres, provides nourishment for that developing marrow. And so at the 32 cell stage, the fluid enters it, collects between the blastomeres, and it reorganizes them around a large fluid-filled cavity, which is called a blastocyst cavity. Once this cavity is formed, the developing mass is now called a blastocyst. So though it is now hundreds of cells, the blastocyst is still staying about the same size as that original zygote. During the formation of this blastocyst, two distinct cell populations arise, the embryoblast and the trophoblast. The embryoblast, or inner cell mass, is located internally and eventually develops into the embryo. The trophoblast is the outer superficial layer of the cells, and that's what forms that spear-like wall of the blastocyst. And it will basically end up developing into the outer chorionic sac that surrounds the fetus and the fetal portion of the placenta. This is the site of exchange of nutrients and waste between the mother and the fetus. Now, around the fifth day after fertilization, the blastocyst hatches, okay, from the zona pellucidum by digesting a hole in it with an enzyme, and then squeezing through the hole. And the shedding from this zone is necessary because it then allows it to permit the next step, okay, implantation or attachment into the vascular glandular endometrial lining of the uterus. So the blastocyst remains free within the uterine cavity, and that's about 2 days before it attaches the uterine wall. At this time, the endometrium is in the secretory phase, so about 6 days after fertilization, the blastocyst loosely attaches to the endometrium. This is a process known as implantation. And as the blastocyst implants, usually in either the posterior portion of the fundus or the body of the uterus, it orients with the inner cell mass toward the endometrium. About 7 days after fertilization, the blastocyst attaches to the endometrium more firmly, and endometrial glands in the vicinity start to enlarge, and the endometrium becomes more vascularized, right, forming new blood vessels. Now, the blastocyst eventually secretes enzymes and burrows into the endometrium and becomes officially surrounded by it. Following implantation, the functional layer of the endometrium is known as the ducta. Now, this separates from the endometrium after the fetus is delivered, and much as it does in just normal menstruation processing. But different regions of this are named based on their positions relative to the site of the implanted blastocyst. The basal ducta is the portion of the endometrium beneath the implanting embryo, and it provides large amounts of glycogen, lipids for the developing embryo and the fetus. And later, it actually becomes a maternal part of the placenta. The capsular ducta is the portion of the endometrium that will cover the embryo after it implants in the endometrium and embryo in the uterine cavity, and the parietal is the remaining modified endometrium that lines the non-invoided areas of the rest of the uterus. So as the embryo and the later the fetus enlarge and push into the uterine cavity, the capsular ducta becomes thin and it eventually disappears as the enlarged fetus fills the uterine cavity and pushes against the surrounding parietal ducta. So by about 27 weeks, the capsular degenerates and it disappears. About 8 days after fertilization, the trophoblast develops into two layers in the region of contact between the blastocyst and the endometrium. These are syncytioblast that contains no distinct cell boundaries, and the cytotrophoblast between the embryoblast and the syncytioblast that is composed of distinct cells. The two layers of the trophoblast become part of the corona, which is one of the fetal membranes, and they undergo further growth. Now, during implantation, the syncytioblast secretes enzymes that enable the blastocyst to penetrate the uterine lining by digesting and liquefying the endometrial cells. Eventually, the blastocyst becomes buried in the endometrium, an inner one-third of the myometrium. Another secretion of the trophoblast is human chorionic gonadotropin, HCG. Now, that has actions very similar to LH. Human chorionic gonadotropin rescues corpus luteum from degeneration and sustains its secretion of progesterone and estrogens. These are the hormones that maintain the uterine lining in a secretory state, preventing menstruation. Peak secretion of HCG occurs about the ninth week of pregnancy, at which this time the placenta is fully developed and produces progesterone and estrogen that continues to sustain the pregnancy. The presence of HCG in maternal blood or uterine is actually the common indicator of pregnancy. Okay, and what's obsessed with the pregnancy test. So, like those of the trophoblast, cells of the embryoblast also differentiate into two layers around 8 days after fertilization, a hypoblast and an epiblast. These are cells of the hypoblast and epiblast together form a flat disc referred to as a bilaminar embryonic disc. So soon a small cavity appears within this epiblast and eventually enlarges to form the amniotic cavity. As the amniotic cavity starts to enlarge, a single layer of squamous cells forms this dome-like roof above the epiblast cells. It's called the amnion. So the amnion forms the roof of the amniotic cavity, and the epiblast forms the floor. Initially, the amnion overlies only the bilaminar embryonic disc, and however, though, the embryonic disc increases in size and begins to fold the amnion and eventually surrounding the entire embryo, creating the amniotic cavity that becomes filled with amniotic fluid. Most amniotic fluid is initially derived from maternal blood, and later the fetus contributes to the fluid by excreting urine into the amniotic cavity. Amniotic fluid serves as basically a shock absorber for the fetus, so it helps to regulate fetal body temperature, prevents the fetus from drying out, adhesions around the skin, and like I like to imagine it as being a test dummy in one of those car accident thingy movies. The amnion usually ruptures, okay, just before birth. It's a fluid that's the basically like a bag of water coming out, okay. Embryonic cells are normally sloughed off into amniotic fluid, and they can be examined in a procedure called amniocentesis, which involves withdrawing some of the amniotic fluid that bathes the developing fetus and then allows us to analyze either fetal cells or dissolved substances that are within the system to confirm, safety and health of the mother and child. Now, on the 8th day after fertilization, cells also at the edge of the hypoblast start to migrate and cover the inner surface of the blastocyst wall. The migrating columnar cells become squamous or flat, and then they form a thin membrane referred to as the extrathymbronic endoblast. Now, together with the hypoblast, the exothalamic membrane forms the wall of the umbilical vesicle, also known as the yolk sac. The former blastocyst cavity during earlier development. Now, as a result, the bilaminar embryonic disc is now positioned between the amniotic cavity and the umbilical vesicle. And since human embryos receive their nutrients from the endometrium, the umbilical vesicle is relatively empty and small. It decreases in size as development progresses. So, nonetheless, the umbilical vesicle has several important functions in humans. It supplies the nutrients to the embryo during the second and third weeks of development. It's the source of blood cells from the third through 6 weeks, contains the first cells or primordial germ cells that will eventually migrate into the developing gonads, differentiate into the primitive germ cells, and form gametes, forms part of the digestive canal, and function as that shock absorber to help with any type of damage that it may ensue. Now, on the 9th day after fertilization, the blastocyst becomes completely embedded in the endometrium. And as the sphincteroplast expands, small spaces called trophoblastic lecma develop within it. By the 12th day of development, the trophoblastic lecma fuse to form a larger interconnecting spaces called the vascular circles or lacunar networks. Endometrial capillaries around this developing embryo become dilated and are now referred to as maternal sinusoids. As these cyclotrophoblasts erodes some of the maternal sinusoids, the endometrial glands and maternal blood and secretions from the glands enter the vascular circles and start to flow through them. Maternal blood is actually both rich in materials for embryonic nutrition, but it also helps with disposing embryonic waste. Now, about the 12th day after fertilization, this extraembryonic mesoblast mesoderm develops these mesoblast cells deriving from the umbilical vesicle and actually forms a connective tissue layer. And that's around the amniotic and the umbilical vesicle. Now, soon a number of large cavities develop in these extraembryonic mesoblasts, which fuse together, forming a single layer cavity called the extraembryonic coleum. The extraembryonic mesoblast, together with these two layers of the trophoblast, form the chorion. The chorion surrounds the embryo and later the fetus, and it eventually becomes the principal embryonic part of the placenta or the structure for exchange of materials between mother and fetus. Now, it also protects the embryo from the immune response. Okay, it secretes proteins that block antibody production by the mother. It promotes the production of T lymphocytes that suppress normal immune response, and it also produces that HCG, which is, we can see an important hormone for pregnancy. The inner layer of this eventually fuses with the amnion. With the development of it, the extraembryonic coleum is now referred to as the chorionic cavity. And by the end of the second week of development, the bilaminar embryonic disc becomes connected to the trophoblast by a band of extraembryonic mesoblasts called the connecting stalk. The connecting stalk is eventually going to become that future umbilical cord. Third embryonic week begins a 6-week period of very rapid development and differentiation. So during the third week, the three primary germ layers are established and they lay the groundwork for the organ development, which occurs from weeks 4 through 8. And the first major event of the third week of development is gastrulation. This occurs about 15 days after fertilization, and this process is bilaminar, so two-layered embryonic discs consisting of epiblast and hypoblast, which then transforms into a trilaminar or three-layered embryonic disc. And this time it has three layers: the ectoderm, mesoderm, and the endoderm. These primary germ layers are the major embryonic tissues from which the various tissues and organs of the body develop, which, if you remember, we talked about a lot of them before.
Gastration involves rearrangement and migration of cells from the epiblast. Okay, the first evidence of gastration is the formation of the primitive streak, which is a faint groove on the dorsal surface of the epiblast that elongates from the posterior to the anterior part of the embryo. The primitive streak clearly establishes the head and tail ends of the embryo, as well as its right and left sides. At the end of the primitive streak, a small group of epiplastic cells form a rounded structure known as the primitive node. Now, following the formation of that primitive streak, cells of the epiblast move inward below the primitive streak and detach from the epiblast, which is known as invagination. Once those cells have invaginated, some of them displace the hypoblast and form the endoderm. Other cells remain between the epiblasts and the newly formed endoderm to form the mesoderm. Now, cells remaining in the epiblast then form the ectoderm, and the ectoderm and endoderm are epithelia, which are composed of those tightly packed cells. The mesoderm is loosely organized connective tissue. As the embryo develops, the endoderm ultimately becomes the epithelial lining of the digestive canal, respiratory tract, and several other organs. The mesoderm gives rise to muscles, bones, and other connective tissues, as well as the peritoneum. The ectoderm develops into epidermis of the skin and the nervous system. Now, about 16 days after fertilization, mesodermal cells from the primitive node migrate toward the head end of the embryo. They form a hollow tube of cells in the midline that's called the notochordal process. And by days 22 to 24, this chordial process becomes a solid cylinder of cells called the notochord. And this structure plays an extremely important role in induction, or the process by which one tissue, or an inducing tissue, stimulates the development of an adjacent unspecialized tissue known as a responding tissue, and allows it to push it into a specialized tissue that it needs to be. An inducing tissue usually produces a chemical substance which influences the responding tissue, and the notochord induces certain mesodermal cells to develop into vertebral bodies using this method. And it also helps to form the nucleus pulse of the intervertebral disc. So also during the third week of development, two faint depressions appear on the dorsal surface of the embryo where the ectoderm and endoderm make contact, but lack of mesoderm between them. The structure closer to the head end is called the orphaneal membrane. It breaks down during the fourth week to connect the mouth cavity to the pharynx and the remainder of the digestive canal. The structure closer to the tail is called the cloacal membrane, which degenerates in the seventh week to form the opening of the anus and urinary and reproductive tracts. And when the cloacal membrane appears, the wall of the umbilical vesicle forms this small vascularized outpatching called the allantois, and this extends into the connecting stalk. In non-mammalian organisms enclosed in an amnion, the allantois is used for gas exchange and waste removal. So because of the role of the human placenta in these activities, the allantois is not a prominent structure in humans. So nevertheless, though, it does function in the early formation of blood and blood vessels, and it is associated with the development of the urinary bladder. So in addition to inducing mesodermal cells to develop in the vertebral bodies, the notochord also induces ectodermal cells over it to form the neural plate. So by the end of the third week, the lateral edge of the neural plate becomes more elevated and from the neural fold. The depressed midregion is now known as the neural groove. So generally, the neural folds approach each other and fuse. This converts the neural plate into a neural tube. This occurs first near the middle of the embryo and then progresses toward the head and tail. The neural tube cells then develop into the brain and the spinal cord. And the process by which this neural plate, neural folds, and neural tube form is all done is called neurulation. Now, as the neural tube forms, some of the ectodermal cells from the tube migrate to form several layers of cells called the neural crest. Neural crest cells give rise to all sensory neurons and prostaglandic neurons of our peripheral nerves, the suprarenal medulla, the melanocytes of our skin, the arachnoid matter and pie matter of the brain, spinal cord, almost all of our skeletal and connective tissue components of our head. But then at about 4 weeks after fertilization, the head end of the neural tubes develop into three enlarged areas called primary brain vesicles or forebrain, mesencephalon, and rhombencephalon. This is midbrain or hindbrain, respectively. At about 5 weeks, the prosencephalon develops into secondary brain vesicles called the telencephalon and diencephalon, and the rhombencephalon develops into secondary brain vessels called the metencephalon and myelencephalon. The areas of the neural tube adjacent to the myelencephalon develop into the spinal cord, parts of the brain that develop from the various brain vesicles, which can contribute to later functions. Now, by about the 17th day after fertilization, the mesoderm adjacent to the notochord and the neural tube forms paired longitudinal columns of paraxial mesoderm. The mesoderm lateral to the paraxial mesoderm forms paired cylindrical masses, which are called intermediate mesenchyme. Now, the mesoderm lateral to the intermediate mesenchyme consists of a pair of flattened sheets. These are referred to as lateral plate mesoderm. The paraxial mesoderm soon segments into a series of paired cube-shaped structures. Those are called somites. And by the end of the 5th week, we got about 40-ish, 42, 44 pairs of somites, which are present. And the number of them which develop over a given period is correlated to the age of the embryo. So it's kind of a way to track. Now, each somite differentiation into two distinct regions, whether it be a dermatome and a sclerotome. The dermatome further differentiates into a dermatome that will contribute to the formation of subcutaneous tissue in dermis and a myotome, which will give rise to all the skeletal muscles of the trunk and limbs. Sclerotome give rise to the vertebrae and the ribs. In the 3rd week of the development, small spaces appear in the lateral plate mesoderm, and these spaces soon merge to form a large cavity, which we know as the intraembryonic ileum. This cavity splits the lateral plate mesoderm into two parts, called splanchnopleurotic mesenchyme and somatophoric mesenchyme. Splanchnophoric mesenchyme forms the heart and the visceral layers of the serous pericardium, the blood vessels, the smooth muscle and connective tissues of the respiratory and digestive organs, and the visceral layer of the serous membrane of the peritoneum. Somatophoric mesenchyme gives rise to the bones, ligaments, blood vessels, connective tissues of the limbs, parietal layer of the serous membrane of the pericardium, the peritoneum. Now, at the beginning of the 3rd week, angiogenesis, or the formation of blood vessels, begins, specifically in the extraembryonic mesoblast, in the umbilical vesicle connecting the stalk and the chorion. This early development is necessary because there's insufficient yolk in the umbilical vesicle and ovum to provide adequate nutrition for the rapidly developing embryo. Angiogenesis is initiated when mesodermal cells differentiate into hemangioblasts, and then these then develop into cells called angioblasts, which aggregate to form isolated masses of cells referred to as blood islands. Spaces soon develop in the blood islands and they form lumens of blood vessels. So some angioblasts arrange themselves around each space to form the endothelium and the tunic layers of the developing blood vessels. And as the blood islands start to grow and fuse, they soon actually form an extensive system of blood vessels all the way throughout the embryo. About 3 weeks after fertilization, blood cells and blood plasma begin to develop outside the embryo from hemangioblasts, specifically in the blood vessels and their walls of the umbilical vesicle, as well as the cones. These then develop into multipotent stem cells that form blood cells, and the blood formation begins within the embryo at about the 5th week in the liver and the 12th week in the spleen, red bone marrow, and the thymus. The heart forms from the splanchnophoric mesenchyme in the head end of the embryo on days 18 and 19. This region of mesodermal cells is called the cardiogenic mesenchyme. In response to induction signals from underlying endoderm, these mesodermal cells form a pair of heart primordial tubes. The tubes fuse to form a single heart tube, and by the end of the third week, the heart tube bends on itself and becomes S-shaped and begins to beat. It then joins blood vessels and other parts of the embryo, and that then connects the stalk, the chorion, and so on. As embryonic tissue invades the uterine wall, maternal uterine vessels are eroded, and maternal blood fills spaces called trophoblastic ligands. Within the invading tissue, by the end of the second week of development, chorionic villi begin to develop, and these finger-like projections consist of chorion surrounded by sphinctoblast and cytoblast. That projects into the endometrial wall of the uterus. Now, by the end of the third week, these blood capillaries develop into the chronic villi, and the blood vessels in the chronic villi connect to the embryonic heart by the way of the umbilical arteries and umbilical vein through that connecting stalk, which will eventually become that umbilical cord. The embryonic blood capillaries within the coronal villi can project into the trophoblastic lacunae, which unite to form intervillous space. That then bathes the chorionic villi with maternal blood. So as a result, the maternal blood, the chorion-covered fetal blood vessels, however, note that maternal and embryonic blood vessels do not join. Okay, and the blood they carry does not mix. Okay, instead, it's just oxygen nutrients in the blood of the mother, intravillous spaces, and spaces between the chorionic villi. Okay, they diffuse across the cell membrane into the capillaries of the villi. Waste products such as carbon dioxide and everything like that, they go in the opposite direction. Okay. Placentation is lastly the process that's forming the placenta. Okay, the site of exchange of nutrients and waste between mother and embryo. The placenta also produces hormones needed to sustain the pregnancy. And placenta is unique, okay, because it develops from two separate individuals, the mother and the embryo. So by the beginning of the 12th week, the placenta has two distinct parts: fetal portion formed by the colonic villi of the chorion, and the maternal portion formed by the basal layer of the endometrium of the uterus. When fully developed, the placenta is shaped like a pancake. Functionally, the placenta allows oxygen and nutrients to diffuse from maternal blood into fetal blood, while carbon dioxide and waste diffuse from fetal blood into maternal blood. The placenta also obviously is a protective barrier, and it's because most microorganisms cannot pass through it. Some viruses can, okay, such as AIDS, measles, chickenpox, things like that, as well as some larger things like drugs can, okay, alcohol, things like that. That's why I tell you not to drink. But of course, positive things like carbohydrates, proteins can also assist with this as well. And the actual connection between placenta and embryo and later the fetus is through the umbilical cord, okay, which develops from connecting stalk and is usually about like an inch long or inch wide, sorry. Actually 24 inches in length. It's pretty big. The umbilical cord consists of two umbilical arteries that carry deoxygenated fetal blood to the placenta, and one umbilical vein that carries oxygen and nutrients acquired from the mother's intravillous spaces into the fetus and supporting mucous connective tissue, which is derived from the lantus. And a layer of amnion surrounds the entire umbilical cord and gives it its shiny appearance. Now, in some cases, the umbilical vein is used to transfuse blood into a fetus or to introduce drugs, various medical treatments via, what is needed for the mother and whatnot. In about 1 in 200 newborns, only one of the two umbilical arteries is present in the umbilical cord. It may be due to failure of the artery to develop or degenerate of the vessel early in development, but nearly 20% of these infants with this condition actually can lead to developing cardiovascular defects. But after the birth of the baby, the placenta detaches from the uterus and is therefore termed the afterbirth. And at this time, the umbilical cord is tied and then severed. And the small portion of the cord remains attached to the infant and it begins to wither and falls off, usually within 12 to 15 days after birth. The area where the cord was attached becomes covered by a thin layer of skin and scar tissue forms, and the scar is the umbilicus or navel. Okay. Now, it's interesting. Pharmaceutical companies actually use human placenta as a source of hormones, drugs, blood, and some actually portions of the placenta are used for burn coverage. Okay, the placenta and the umbilical cord veins actually can also be used in blood vessel grafts as well. And the cord blood frozen to provide future source of pluripotent stem cells, so such as repopulating red bone marrow following radiotherapy for cancer.
4 through 8 weeks of development are very significant in embryonic development because all major organs appear during this time. The term organogenesis refers to this formation of the body organs and systems at this key time point. And by the end of the 8th week, all of the major body systems have begun to develop. Although their functions for the most are minimal, organogenesis requires the presence of a lot of things, and specifically blood vessels, because we need to get the nutrients, the oxygen to the organs. However, recent studies suggest that blood vessels, while play a significant role in organogenesis even before blood beginning to flow with them, whereas endothelial cells of blood vessels can provide some type of developmental signal, like a secreted substance, which has the cells kind of talk to each other to support organogenesis even without the presence of blood. During the 4th week, though, at fertilization, the embryo undergoes very dramatic changes in shape and size, nearly tripling its size. It is essentially covered from or converted from a flat two-dimensional trilaminar embryonic disc to a three-dimensional cylinder, which is a process known as embryonic folding. The cylinder consists of endoderm in the center, okay, gut, ectoderm on the outside, epidermis, and mesoderm in between. The main force responsible for embryonic folding is the different rates of growth of various parts of the embryo, especially the rapid longitudinal growth of the nervous system, of the nervous tube. Now, folding in the median plane produces a head, and fold in a tail in the horizontal plane results in two lateral folds. So overall, due to the folding, the embryo kind of curves in on itself and forms a C shape. The head fold brings the developing heart and mouth into the eventual adult positions. The tail fold brings the developing anus into its eventual adult position. And the lateral folds form as the lateral margins of the trilaminar embryonic disc bend ventrally. As they move toward the midline, the lateral folds incorporate the dorsal part of the umbilical vesicle into the embryo as a primitive gut, which is the forerunner of a digestive canal. The primitive gut differentiates into an anterior foregut and an intermediate midgut and a posterior hindgut. The fates of the foregut, midgut, and hindgut, as you recall, in the orphangeal membrane is located in that head of the embryo. It separates the future pharyngeal or throat region of the foregut from the stomodium and the future oral cavity. Because of head folding, orphangeal membrane moves downward in the foregut and stomodium moves closer to the final positions. When the orphangeal membrane ruptures during the 4th week, pharyngeal region of the pharynx is brought into contact with the stomata. In a developed embryo, the last part of the hindgut expands into a cavity which is called the cloaca. On the outside of the embryo is a small cavity in the tail region which is called the proctoduum. Separating the cloaca from the proctoduum is the cloacal membrane. Now, during embryonic development, the cloaca divides into a ventral urogenital sinus and a dorsal and a rectal canal. As a result of tail folding, the cloacal membrane moves downward, and the urogenital sinus and a rectal canal and proctoderm move closer to their final positions. When the cloacal membrane ruptures during 7th week of development, the urogenital and anal openings are created. In addition to embryonic folding, development of somites, and development of the neural tube, 4 pairs of pharyngeal arches or bronchial arches begin to develop on each side of the future head and neck regions. This is specifically during the 4th week.
These four paired structures begin their development on the 22nd day after fertilization and form swellings on the surface of the embryo. Each pharyngeal arch consists of an outer covering of ectoderm and an inner covering of endoderm, with mesoderm in between. And within each of these arches, there is an artery, a cranial nerve, cartilaginous skeletal rod, which supports the arch, and skeletal muscle tissue, which attaches to and moves the cartilage rods. On the ectoderm surface of this region, each arch is separated by a groove called a pharyngeal groove. And these grooves meet corresponding balloon-like outgrowths of the endodermal lining called the pharyngeal bronchial pouches. This is where the pharyngeal groove and pouch meet to separate the arches, and the outer ectoderm of the cleft contacts the inner endoderm of the pouch, and there's actually no mesoderm finally in between. So just as the somite gives rise to specified structures of the body wall, each arch, groove, and pouch give rise to specified structures in the head and neck. Each arch is a developmental unit and includes a skeletal component, muscle, nerve, and blood vessels. And in the human embryo, there are four obvious arches. Each of these arches develop into a specific and unique component of the head and neck region. So, like the first one is often called a mandibular arch because it actually forms the jaw. The first sign of developing ear is a thickened area of the ectoderm, an aortic placoid or future internal ear, which can be distinguished about 22 days after fertilization. A thickened area of ectoderm called the lens placoid, which will eventually become the lens of the eye, will also appear around this time. By the middle of the fourth week, the upper limbs will then also begin their development as outgrowths of mesoderm covered by ectoderm, which is called the upper limb buds. By the end of the fourth week, the lower limb buds develop, and the heart also forms a distinct projection on the ventral surface of the embryo called the heart prominence. At the end of the fourth week, the embryo will also have a distinctive tail. During the fifth week of the development, there's a very rapid development of the brain, so growth of the head is considerably increased. And by the end of the sixth week, the head grows even larger relative to the trunk, and the limbs show substantial development. In addition, the neck and trunk begin to straighten, and the heart is now four-chambered. By the seventh week, the various regions of the limbs become distinct, and the beginnings of digits start to appear. And at the start of the eighth week, which is the final week of the embryonic period, the digits of the hands are short and webbed, and the tail is shorter but still visible. The eyes are finally open, and the auricles of the ears are visible. By the end of the eighth week, all regions of limbs are apparent. The digits are distinct. There's no longer webbed due to removal of the cells via apoptosis, and the eyelids come together. Okay, they may fuse. The tail does disappear finally, and external genitalias begin to differentiate. And now we are seeing human characteristics. During the fetal period, which if you remember is ninth week until birth, tissues and organs that develop during that entire embryonic period grow and differentiate. So very few new structures will appear during the fetal period. But the rate of the body growth is quite impressive, okay, especially during the second half of the intrauterine life. So, for example, the last 2.5 months of the life, half of the full term weight is added. So at the beginning of the fetal period, the head is half the length of the body. By the end of the fetal period, the head side is only 1/4 the length of the body. During the same period, the limbs also increase in size from 1/8 to 1/2 the fetal length, and the fetus is also less vulnerable to damaging effects of drugs, radiation, and microbes than it was in this sensitive embryo period. Exposure of developing embryo or fetus to certain environmental factors, though, can of course damage it. Okay, can damage the developing organism or at times can even cause death. These are known as teratogens, and that's just any agent that can influence the causes developmental change, defects, and even possibly death. Okay, and there are several ones, such as chemicals and drugs. Okay, it's probably one of the most common ones because placenta is not an absolute barrier, okay, between maternal and fetal circulations. A drug or chemical that is dangerous to an infant, it's got to be considered potentially dangerous to the fetus when given to the mother. So
That's why you get the consult your doctor if you're considering being pregnant, if you are pregnant, things like that. And probably one of the most common ones is alcohol. It's like the number one fetal terrogen. And exposure to even just a small amount of alcohol can result in fetal alcohol syndrome, which is one of the most common causes of mental retardation and the most common preventable cause of birth defects in the United States. Okay. And it's all associated with slow growth, uh, messing with the developmental changes, especially even after birth characteristics being slowly developed, things like that. But of course, there's others, viruses, hepatitis B, C, pesticides, industrial chemicals, pollution, antibiotics. The list can go on and on and on and on. This is why being exposed and aware of your certain environment while caring or planning on caring is quite important. So, okay, dang, stuff can go wrong, right? But we have tests to see if there is things that are going wrong. There's several tests that we can do. We can do what is known as ultrasonography, which is just trying to look and use a common method of ultrasound to get this diagnosis, to whether to determine the accurate fetal age, if there's any change in development length, development size, even confirming pregnancy, evaluating, fetal vitality, things like that. And it's pretty easy. It's literally just doing an ultrasound. Okay. Another is an amniocentesis, which is just withdrawing some of the amniotic fluid, okay, with inside and surrounding the developing fetus. This allows us to see any sloughed off fetal cells, to see if there's any changes. This we can get DNA to test for things like Down syndromes, hemophilia, Tay-Sachs disease, sickle cell disease, seeing if there's any changes in the pH of this fluid. Okay, that may be affecting the development. It's quite easy. It's just syringe, and it's collecting a sample. Now, obviously, the mother's going to have some changes as well, right? This is a feed off kind of relationship. Okay, they're both impacting one another. And so, one of the most major things impacted during and years after are hormones of the mother. So during the first 3 to 4 months of pregnancy, the corpus luteum in the ovary continues to secrete progesterone and estrogens. Okay, that maintains the lining of the uterus during pregnancy and preparing the mammary glands to secrete milk. Now, the amounts secreted by the corpus luteum, however, are only slightly more than those produced after ovulation in a normal menstrual cycle. From the third month through the remainder of the pregnancy, though, the placenta itself provides the high levels of progesterone and estrogens required. So, as we saw previously, okay, the chorion of the placenta secretes the human clonic gonadotropin, HCG. So, secreting it into the blood, and in turn, HCG stimulates the corpus luteum to produce progesterone and estrogen, an activity required to prevent the menstruation and for the continued attachment of the embryo and fetus to the lining of the uterus. By the 8th day after fertilization, HCG can be detected in blood, and if you remember, that is what it's looked for, okay, when we're taking our pregnancy test. Okay, so peak secretion of the HCG will occur about the 9th week of pregnancy. During the 4th and 5th months, HCG level decreases sharply and then levels off all the way until childbirth. The chorion begins to secrete estrogens after the first 3 or 4 weeks of pregnancy and progesterone by the 6th week. And these hormones are secreted in increasing quantities until the time of birth. So by the 4th month, when the placenta is fully established, the secretion of HCG is generally reduced, and the secretions of the corpus luteum are no longer essential. A high level of progesterone ensures that the uterine myometrium is relaxed and that the cervix is tightly closed. After delivery, estrogens and progesterone in the blood decrease to the normal levels. Relaxin is a hormone that's produced first by the corpus luteum of the ovary and later by the placenta. And it increases the flexibility of the pubic and the ligaments of
the sacralic and sarcoglial joints. This is what helps to dilate the uterine cervix during labor. So both of these actions are important because they help to ease the delivery of the baby and have a successful delivery. A third hormone that's produced by it is, or by the placenta, is human chorionic somatotropin, or HCS. It's also known as human placental lactogen, HPL. The rate of secretion of this increases in proportion to the placental mass, and it reaches maximum levels after its 32 weeks, and it remains pretty constant after that. And so it's thought that this helps prepare the mammary glands for lactation and enhances maternal growth by increasing protein synthesis and regulates certain aspects of metabolism, not just in the mother, but also in the fetus. So, for instance, HCS decreases the use of glucose by the mother and promotes the release of fatty acids from her adipose tissue, making more glucose available to the fetus. Now, the hormone most recently found to be produced by the placenta is corticotropin releasing hormone, or CRH. This, in non pregnant people, is secreted only by neurosecretory cells in the hypothalamus. It is now thought to be part of the clock that establishes the timing of the birth. So secretion by the placenta begins at about 12 weeks and it increases enormously towards the end of pregnancy. So women who have higher levels of the CRH early in their pregnancy are more likely to deliver prematurely. Those who have low levels are more likely to deliver after their due date. CRH from the placenta has a second important effect too, and it increases the secretion of cortisol, which is needed for the maturation of the fetal lungs and the production of surfactant. During the pregnancy, a lot of other physiological changes occur. Okay, so for instance, near the end of the third month of pregnancy, the uterus occupies most of the pelvic cavity. So as the fetus continues to grow, the uterus extends higher and higher into the abdominal cavity. Towards the end of a full-term pregnancy, the uterus fills nearly the entire abdominal cavity, reaching above the costal margin nearly to the xiphoid process of the sternum, and it pushes the maternal intestines, liver, stomach superiorly, and it elevates the diaphragm and widens the thoracic cavity. So pressure on the stomach can cause some issues and may even force the stomach contents superiorly into the esophagus, which is why a lot of pregnant women have heartburn, for instance. And in the pelvic cavity, it has so much compression on the ureters or the urinary bladder, which gives you the more, I need to pee all the time kind of feeling for pregnant women. Other pregnancy-induced physiological changes occur as well. So weight gain, okay, obviously we're growing a human, right? We're going to gain some weight from that. The amniotic fluid, the placenta, the total body water is increased, the uterine is enlarged. So there's a lot of weight gain going through this, okay? There's a lot of increased storage of macromolecules, proteins, fats, triglycerides, minerals, there's breast enlargement, okay, and that's because of preparation for lactation, and commonly lower back pain, okay, due to lordosis or hollow back. Now, several changes occur in the maternal cardiovascular system. Stroke volume, for instance, increases and cardiac output rises. This is all due to increased maternal blood flow to the placenta because we need to basically get the essentials for metabolism responses to the fetus, right, to help it grow. So because of this, we need to compensate for that. So as a result, heart rate's going to increase, blood volume's going to increase, and it's all mostly during the second half of the pregnancy. And these increases are necessary. All right, we have to meet that initial demands of the fetus for nutrients and oxygen. So when a pregnant woman is lying on her back, the enlarged uterus may compress the aorta, resulting in diminished blood flow to the uterus. So compression of the inferior vena cava also decreases venous return, which leads to the edema in the lower limbs and may produce some varicose veins. So compression of the renal artery can lead to renal hypertension. Respiratory functions also alter during pregnancy. Okay, again, it's about meeting the added demand of the fetus, specifically the oxygen demands. All right, tidal volume can increase. Expiratory reserve volume can be reduced. Functional residual gas speed can be decline. It can decline as well. Minute ventilation, or the total volume of air inhaled and exhaled each minute, can increase. Airway resistance in the bronchial tree can decrease. and total body oxygen consumption can increase. And because of all this kind of occurring, a lot of time these pregnant women go through what's known as dyspnea, which is just difficulty breathing. Okay. There's obviously some other physiological features that can cause that, as well as the compression onto the diaphragm. The digestive system also undergoes some changes, okay, such as an increase in appetite. Again, meeting the nutritional demands of the fetus. So you're going to eat more. You're going to need the calories, the proteins, and so forth. A general decrease in digestive canal motility causes constipation, delay in gastric emptying. It produces some nausea, vomiting, and heartburn. So common things you would think of with someone who's pregnant. We kind of talked about as well, but there's going to be increased pressure on the urinary bladder. Okay, so that enlarging uterus, it can produce increased urinary symptoms. Okay, more frequency, more urgency of urination. Sometimes it even feels like you have to go to the bathroom, but you can't. Okay, so it increases the renal plasma flow and glomerular filtration as a result, because we need to get rid of more of the extra waste that's produced by the mom, but also the fetus. Changes in the skin during pregnancy are more apparent in some women than in others, so not all can see this. Now, some women experience increased pigmentation around the eyes and cheekbones in a mask-like pattern, and in the areola of the breast, and then a horizontal line along the lower abdomen called the line intergen. Now, stretch marks often are formed, okay, because the abdomen is extending. There is some weight gain, okay, so this results in some stretch marks, okay. Changes in the genital system, as we would expect, there's going to be changes, right? And it's including edema, increased vascularity of the vulva, increased flowability and vascularity of the vagina. The uterus increases from its non-pregnant mass, mostly because of the hyperplasia of muscle fibers and the myometrium in early pregnancy and the hypertrophy of the muscle fibers during the second and third trimesters. But only a few changes in early pregnancy affect the ability of a woman to exercise. It can really impact it. And it's a lot of it is, well, one, it's pretty hard, right, for someone to go be through this and exercising in general. So there is a lot of mental kind of stuff with it, but they may tire more easily. Okay, they have morning sickness, may interfere with regular exercise. As the pregnancy progresses, weight is gained, posture changes. There's back pain. Energy that is needed is hard to get. Okay, you can't eat so much, right, and sleep so much. Certain inflammation and other issues may occur, so you'll have joint pain similar to, arthritis, things like that, which brings in its difficulties of exercising while pregnant, even if it is known to be so helpful. Okay, so we finally get to the end, right? Okay, baby's developed. We've gone through the egg coming together with the sperm, the implantation, the development, the folding. The mother has had her hormones change. She's grown in the belly. She's tired, and it's time to go. Labor is here. So labor is that process by which the fetus is just expelled from the uterus through the vagina. Okay, you've heard as giving birth. Okay, so obviously, this is going to cover vaginal birth. There's more than vaginal birth. There's C-sections. Okay, so more of the surgical birth, commonly with, women have issues with baby issues, and for instance, like that. This, we're going to talk about vaginal. Okay. Now, the onset of labor is determined by complex interactions. Okay, several things are going to interact with one another. Placental and fetal hormones, one of the big ones. So because progesterone inhibits uterine contractions, labor cannot take place until effects of progesterone are diminished. So toward the end of gestation, the levels of estrogens in the mother's blood rise sharply. This produces changes that overcome the inhibiting effects of progesterone, and the rise in estrogen results from increasing secretion by the placenta, or the corticotropin releasing hormone, which stimulates the anterior pituitary gland of the fetus and secretes ACTH, or adrenocorticotropic hormone. In turn, ACTH stimulates the fetal suprarenal gland to secrete cortisol and dehydroandrosten, DHEA. the major suprarenal androgen. The placenta then converts this into an estrogen. Okay, and high levels of estrogens cause the number of receptors for the oxytocin on uterine muscle fibers to increase and cause uterine muscle fibers to form gap junctions with one another. Oxytocin released by the posterior pituitary stimulates uterine contractions, and relaxin from the placenta assists it by increasing the flexibility of the region, helping to dilate the uterine cervix. Estrogen also stimulates the placenta and releases prostaglandins. These induce production of enzymes that digest collagen fibers in the cervix, again causing it to soften. The goal here is to make labor easy. Control of labor contractions during this, it's all by positive feedback cycle. Okay, contractions of the uterine myometrium forces the baby's head or body into the cervix and descends the cervix or stretches. Okay, stretch receptors in the cervix send nerve impulses to neurocirculatory cells in the hypothalamus, causing them to release oxytocin into blood capillaries of the posterior pituitary gland. Oxytocin then is carried by the blood to the uterus, where it stimulates the myometrium to contract more forcefully. As the contractions intensify, the baby's body stretches. Okay, the cervix still more, and the resulting nerve impulses stimulate the secretion of yet more oxytocin. You're building up on the oxytocin. With birth of the infant, the positive feedback cycle is broken because cervical distension suddenly loosens. Okay. Uterine contractions occur in waves. Okay, so very similar to peristaltic waves of the gastrointestinal tract. They start at the top of the uterus and move downward, eventually expelling the fetus. And true labor begins when uterine contractions occur at regular intervals, usually producing pain. So as the interval between the contractions shorten, the contractions intensify. And another symptom of this true labor in some women is localization of the pain in the back that is intensified by walking. And the most reliable indicator of true labor is dilation of the cervix and the show or discharge of blood-containing mucus in the cervical canal. Now, in false labor, pain is felt in the abdomen of irregular intervals, but it does not intensify, and walking does not alter it significantly. So there's no show and no cervical dilation. And true labor, once it gets there, it's divided into three stages. Okay, first, the stage of dilation. That's the time from onset of labor to the complete dilation of the cervix. Okay, the stage, which typically lasts 6 to 12 hours. Okay, it's got regular contractions of the uterus, usually a rupturing of the amniotic sac, and complete dilation. Complete dilation is considered 10 centimeters. If the amniotic sac does not rupture spontaneously, the doctors do go forth and they rupture this intentionally. Okay. Now on to the next stage is stage of expulsion. Now the time from complete cervical dilation to delivery of the baby is the stage of expulsion. Okay. That tire pushing process expelling. Okay. Now after the stage of expulsion, we get to the placental stage. This time, 3 to 5 minutes or up to an hour or more after delivery into the placenta or after birth, it's sometimes referred to. It's expelled by the powerful contractions of the uterine. Okay, these contractions also constrict blood vessels that were torn during delivery. They reduce the likelihood of hemorrhaging. Okay, so as a rule, labor lasts longer with first babies, so typically about 14 hours. For women who have previously given birth, the average duration of labor is about 8 hours. So although the time varies enormously among births, because fetus may be squeezed through the birth canal, cervix, vagina for up to several hours, the fetus is stressed during childbirth. So the fetal head is compressed. The fetus undergoes some degree of intermittent hypoxia due to compression of the umbilical cord and the placenta during the uterine contractions. So in response to this stress, the fetal adrenal medulla secrete very high levels of epinephrine or norepinephrine. That's that fight or flight hormone. Much of the protection against the stressors of birth, as well as preparation of the infant surviving this extreme life, is provided by those hormones. Okay, and among other functions, epinephrine and norepinephrine clear the lungs, and they alter the physiology and readiness for breathing air, mobilizing readily usable nutrients for cellular metabolism, and promoting the flow of blood to the brain and heart. Now, Following the delivery of the baby, okay, the placenta is a 6-week period during which the maternal reproductive organs and physiology return to pre-pregnancy state. This period is called the puerperium. It's through a process of tissue catabolism. The uterus undergoes remarkable reduction in size. All right, so it goes back to mostly back to its normal size where it began. Okay, and this is called involution. Now, this especially has an impact in lactating women, where the breast does change in size. The breast and the mammary gland structures change as well. Once breastfeeding is done, a similar process is undergone, known as involution, where the breast returns back to a similar kind of structure that it used to be. Okay. Now, the cervix after this loses its elasticity and regains its pre-pregnancy firmness. So for 2 to 4 weeks after delivery, women have a uterine discharge called lochia, which consists initially of blood and later of serous fluid that's derived from former site of the placenta. It's completely common. It's just the body returning back to its, normal self. So during pregnancy, the embryo, okay, and later the fetus, is totally dependent on the mother for its existence. The mother supplies the fetus with oxygen, nutrients, it eliminates all its waste like carbon dioxide, protects against the shocks, the temperature change, it provides a form of an immune system by antibodies, fighting off certain harmful microbes. Okay, and at birth, a physiologically mature baby becomes much more self-supporting and the newborn's body systems must make some intense adjustments. Now, the most dramatic changes occur in respiratory and cardiovascular systems. And the reason that the fetus depends entirely on the mother for obtaining oxygen and eliminating carbon dioxide is that the fetal lungs are either collapsed or partially filled with amniotic fluid when they're developing. The production of surfactant begins by the end of the 6 month of development. Because the respiratory system is fairly well developed, at least 2 months before birth, premature babies delivered at 7 months they're pretty able to breathe and cry. But after delivery, the baby's supply of oxygen from the mother ceases, and any amniotic fluid in the fetal lung is absorbed. Because carbon dioxide is no longer being removed, it builds up in the blood. A rising CO2 level stimulates the respiratory center in the medulla oblongata, causing the respiratory muscles to contract, and then the baby to draw his first breath. And because the first breath is unusually deep, as the lungs contain no air, the baby also exhales vigorously and naturally cries. So a full-term baby may breathe 45 times a minute for the first 2 weeks. Okay. Breathing rate, however, does gradually decline until it approaches more of a normal rate of breaths per minute. So after the baby's first breath, okay, the cardiovascular system must make some several adjustments. Closure of the foramen ovule between the atria of the fetal heart, which occurs shortly after birth, diverts deoxygenated blood to the lungs for the first time. And the foramen ovule is closed by two flaps of septal heart tissue that fold together and permanently fuse. The remnant of the foramen ovule is the fossils. Once the lungs begin to function, the ductus arterius shuts off due to contractions of smooth muscle in its wall, and it becomes the ligamentum arteriosum. The muscle contraction is most likely probably mediated by the polypeptide bradykinin that's released from the lungs during the initial inflation. Ductus arterius generally begins to close about a day after birth. Usually it's completed by 21 days after birth. However, prolonged incomplete closure results in some patient issues with blood flow. Now, after the umbilical cord is tied off and severed and blood no longer flows through the umbilical arteries from the mother, they fill with connective tissue and become the medial umbilical ligaments. So the umbilical vein then becomes the ligamentum teres, or the round ligament of the liver. And in the fetus, the ductus venous connects the umbilical vein directly with the inferior vena cava, allowing blood from placenta to bypass the fetal liver. When the umbilical cord is severed, the ductus venous collapses, and the blood from the visceral of the fetus flows into the hepatic portal vein to the liver and then via the hepatic vein to the anterior vena cava. And the remnant of the ductus venous becomes the ligamentum venous. So at birth, an infant's pulse changes, okay, from about 100 tonning to 160 beats per minute, and sometimes even higher to about 180. So after birth, oxygen use needs to increase. So that stimulates an increased rate of red blood cell and hemoglobin production. The white blood cell count at birth is also very high. It can be twice, if not three times as high as normal levels. Okay. However, this is a quick adjustment. So over time, over a couple days, all these things they will return slowly to more of a quote unquote normal rate. Lactation is the production of milk from the mammary gland. Okay, and it's a principal system to get the nutrients to the newly born baby, right? And a hormone that is commonly used to promote milk production is prolactin, okay, which is secreted from our anterior pituitary gland. Now, even though prolactin levels increase as pregnancy progresses, no milk production occurs because progesterone inhibits the effects of prolactin. Now, after delivery, though, the levels of estrogen and progesterone in the mother's blood decrease, and so the inhibition is removed. The principal stimulus in maintaining prolactin secretion during lactation is the sucking action of the infant. And the suckling initiates nerve impulses from the stretch receptors and the nipples all the way to the hypothalamus. The impulse decreases hypothalamic release of prolactin inhibiting hormone and increases the release of prolactin releasing hormones. So more prolactin is released by the anterior pituitary as a result. Now, oxytocin also plays a role. Oxytocin causes the release of the milk into the mammary ducts via the milk ejection reflex. So, milk formed by the glandular alveoli of the breast is stored until the baby begins to actively suckle. Stimulation of touch receptors on the nipple initiates sensory nerve impulses that are relayed to the hypothalamus, and in response, secretion of oxytocin from the posterior pituitary increases. Now, carried by the bloodstream to the mammary glands, oxytocin stimulates contraction of the myoepithelial cells, or smooth muscle-like cells, that surround the glandular alveoli and ducts. So the resulting compression contraction moves the milk from the glandular alveoli into the mammary ducts, where then it can be suckled. Okay, and this process is the milk ejection. Even though the actual ejection of milk does not occur for about a minute after nursing begins, some milk stored in lactiferous sinuses near the nipple is available during latent periods, and stimuli actually other than suckling can also impact this. Sometimes even hearing the baby cry, touching genitals of the mother, things that lead to the oxytocin or love drug being released can contribute to a milk ejection. Okay. Now, the suckling stimulation that produces the release of oxytocin can also make an impact by the release of the inhibitor progesterone, and this results in an increased secretion of prolactin, which maintains its lactation as well. During late pregnancy and the first few days after birth, the mammary glands secrete a cloudy fluid called colostrum. Now, although it is not as nutritious as milk, it contains less lactose, virtually no fat. Colostrum serves adequately until the appearance of true milk, which is about the fourth day following birth. And colostrum and maternal milk contain important antibodies. Okay, they protect infants during the first few months of life, which is why, um, you know, being breastfed is so strongly encouraged for mothers that are able to. Now, following birth of the infant, the prolactin level starts to return to the non-pregnant level. So, however, each time the mother nurses, the infant nerve impulses from the nipples to the hypothalamus increases the release of the prolactin releasing hormone, which results in a tenfold increase in prolactin secretion, specifically by the anterior pituitary. That lasts for about an hour. Now, prolactin acts on the mammary glands to provide the milk for the next nursing period, and if this surge of prolactin is blocked by injury or disease, or maybe nursing is discontinued, the mammary glands lose their ability to produce milk in only a few days. Even though milk production normally decreases considerably within 7 to 9 months after birth, it can continue for several years, though, actually, and it's all dependent on keeping up with the nursing, with the breastfeeding, because after you stop, it kind of drops significantly pretty quick. Now, lactation often blocks ovarian cycles for the first few months as well. So, if the frequency of a suckling is about 10-ish times a day, This can lead to a change in your ovarian cycle. This effect is inconsistent, however. And ovulation commonly precedes the first menstrual period after delivery of the baby. So as a result, the mother can never be certain she's not fertile. Breastfeeding is therefore an unreliable birth control measure, and the suppression of ovulation during lactation is believed to occur because during breastfeeding, neural input from the nipple reaches the hypothalamus and causes it to produce neurotransmitters that suppress the release of the gonadotropin releasing hormone. So because of that, they produce more LH and FSH, which will decrease, and ovulation is inhibited. Now, primary benefit though of breastfeeding is the nutrition. Okay, human milk is a sterile solution and it contains a lot of fatty acids, a lot of lactose, a lot of amino acids, minerals, vitamins, water, all that, everything that's ideal for a baby's digestion, brain development, and growth. And it has a couple other benefits, such as supplying beneficial cells. Okay, several types of white blood cells are present in breast milk. You got neutrophils, macrophages, phagocytes that are ingesting microbes in a baby's digestive canal to assist with their immune system. Macrophages that release lysosomes, again, attacking these microbes. The other T cells, B cells that assist in the same way. Beneficial molecules. Okay, so breast milk can contain a beneficial molecule such as a maternal IgA antibody in breast milk, which binds to microbes in the baby's gastrointestinal tract and prevents their movement into other body tissues of the baby. Now, because a mother produces antibodies to whatever disease causing microbes are present in her environment, her breast milk affords protection against a specific infection to which her baby is also exposed in the environment. Now, additionally, two milk proteins bind to nutrients that many bacteria need to grow and survive. B12 binding protein ties up vitamin B12, and lactoferrin ties up iron. Now, a couple other ones, some specific fatty acids that assist with development as well, neurological development, digestive development as well. But the benefits are really there. It's well studied, well known. Of course, there are concerns and issues with some women not being able to do it. And luckily, we have science now that's working on the best kind of way to handle that nowadays. All right, so a lot of this might be a repeat for some of you. We're talking about development. We're talking about sexual reproduction. So obviously, inheritance, genetics was going to come into play somehow. So we have genetic material of the father and a mother. They unite, right? Sperm fusing with secondary oocyte, you get the zygote. Now, there's a reason why we resemble some of our parents' traits because we inherit them. They're passed down from both parents. And so inheritance is literally that. It's just the passage of hereditary traits from one generation to the next. You look like your parents look like your grandparents, and so forth, so forth, so forth. And it is the process by which you acquire your characteristics. Okay, and they may even transmit some of those to your children. And so that's when we get our genotype and phenotype. All right, so if you remember, we have chromosomes, right? We're diploids. So we have two sets of chromosomes, and each of these two homologs contain genes. These genes control our traits, our physical traits. And if one chromosome of a pair contains a gene for body hair, so say it's a gene for body hair in the same position, and alternative forms of a gene that code for the same trait and are at the same location are homologous chromosomes. They're called alleles. Now, one allele of previously mentioned body hair gene might code for a coarse hair, and another might code for the fine hair. A mutation can occur. Okay, and a mutation is just that permanent heritable change in an allele that produces a different variant to the same trait. The relationship of genes to heredity is illustrated, okay, here by examining how alleles that are involved in disorders called phenylketonuria and people with PKU are unable to manufacture certain enzymes. Okay, but here is just an example on how certain um, you know, carriers, non-carriers do have it, can go. And it's your simple Punnett square, which I'm sure some of you have seen before. So by tradition, okay, symbols of the genes written in italics, right? With dominant alleles written in capital letters and recessive allele in a lowercase letter. So person with the same alleles on the homologous chromosomes, either two big B's, two small B's, is said to be homozygous.
for the trait. Now, two big P's, capital P's, is homozygous dominant, and two shorthand P's is homozygous recessive. Now, an individual with different alleles on homologous chromosomes, so big P, little p, is heterozygous. Okay, and this kind of combination is what contributes to the phenotype or the actual, you know, I guess expression of what is being done, the physical expression, outward expression. So, for this instance, in this Punnett square, it would be if they do have the disease, if they do not have the disease. And if it's a dominant gene, then it kind of overpowers those other recessive genes. Okay. So if I had a heterozygote here, so capital P, lowercase p, but it's a dominant P, then that dominant would overpower the recessive, but the only way being kind of beat off is having a lower recessives. Okay. Now, this is commonly Mendelian genetics. All right, dominant, recessive, Punnett squares, easy stuff. Um, not everything follows in this manner. We can get some other stuff. So, like I said, not everything is simple dominant recessive inheritance. We can have some phenotypic expressions of particular gene that can be influenced by which alleles are present, but genes that also might be in the environment. You can be influenced by more than one gene, and that just complicates a lot of things, obviously, because you're applying a new variable. Most genes can influence more than one trait, and other variations like complex inheritance and complete dominance. And complete dominance, for instance, is just neither member of a pair of alleles is dominant over the other, and the heterozygote has a phenotype that's immediate between a homozygous dominant and the homozygous recessive phenotype. So the most common one that you've probably been shown or know is that if you have a rose that is dominant red and recessive white, those come together to make a heterozygous, then the rose can be pink. It's a blend of one of them. Now, the other one is the multiple allele inheritance. And so although a single individual inherits only two alleles for each gene, some genes may have more than two alternative forms. And this is the basis for multiple allele inheritance because one example, for instance, is your ABO blood. Okay, there's four blood types of the ABO group: A, B, AB, and O. And it's an inheritance of six combinations of three different alleles of a single gene that contributes this. And it's the gene I. Now, allele IA produces the A antigen. Allele B produces the B antigen. And I produces neither A nor B. Now, each person inherits two of those genes, specifically two of those alleles. Now, one from each parent that gives rise to the various phenotypes. So, the six possible phenotypes produce four blood types. So, if you have IA and IB are inherited as dominant alleles, and I is inherited as a recessive allele, because an individual with type AB blood has characteristics of both type A and B red blood cells expressed in a phenotype. Alleles IA and IB are said to be codominant. So in other words, both genes are expressed equally. Now, depending on the parental blood types, different offspring may have blood types different from each other. Okay. Now, the other kind of complex, hence its name, complex inheritance that can come into play is that most inherited traits they're not controlled by one gene. Okay, there's other combined effects of several genes, two genes, even some type of environment that comes into play that messes with it. Okay, so it's these combined effects that can change the actual inheritance when it comes to the traits. This is like skin color, hair color, eye color, your height, your metabolic rate, your body build, things like that. One genotype can have many possible phenotypes, and it all depends on things like the environment. Or one phenotype can include many possible genotypes. It's kind of feed in onto one another. It's just like rolling the dice. Okay. Now, when viewed under a microscope, we got 46 human chromosomes, right? Just 46 chromosomes in a normal somatic cells. But we have 23 different pairs, and there's one special pair that we have. Okay, and it's not those autosomes, the other 22. It's our sex chromosomes. And we already kind of talked about this, XX makes a woman, XY makes genetically a male. Okay, and we talked about how it's, the sperm is the one that determines it. Okay, and when a spermicide undergoes meiosis to reduce its chromosome number, it's going to give rise to two different sperm that contain an X and contain a Y. Okay, remember, oocytes, no Y. Okay, so that's why it is the males determining on what the gender is going to be for woman. Okay, now both female and male embryos develop identically until about 7 weeks of fertilization, right? We covered that. That's the way we are designated. Even two different specific duct systems, which for one being destroyed if it's a male, one being destroyed if it's a female, right? And it keeps it going. Okay, so seeing the whole, SRY, sex determining region of the Y, and we don't have it, then it kind of led to a change in genetic material and the stuff that was produced with testosterone. And eventually you got an idea of what if there's other stuff that is linked to these guys, and that's when we got into studies of sex-linked genes or sex-linked diseases. Okay, so you know, color blindness is one of the most common ones. Okay, it's commonly found on the X chromosome, but it's acts kind of like a recessive gene. Okay, so if the only way a woman's gonna have it is both her X's are recessive. However, all these X-linked diseases are commonly expressed more in men, and it's because we don't have the opportunity to have another X there that could possibly mask that disease like colorblind. So if we get an X chromosome that has it, then we're screwed. Okay, we're going to express it because there's nothing over there to mask it. So alongside with this general link of plain inheritance and the 6,000 different ways it can happen, we also have sex-linked inheritance, which is all dependent on what's coming with our X and what's coming with our Y chromosome. All right, well, that is it for this semester. I hope you guys enjoy your break. And let me know if you have any other questions or need anything from me.